National Seminar, Engaging Science for Animal Health Innovation. For today is the theme of Animal Health and Environment. We have 11 speakers for this meeting. In the first morning session, we will have a warm welcome address by Professor Dr. Sathaporn Chitta Palapong, the Dean of Faculty of Veterinary Technology, Kasetsart University. Then we will have the first talk from Associate Professor Dr. Hiroshi Shimoda from Laboratory of Veterinary Microbiology, Yamaguchi University, Japan. At the end of the day, the conclusion will be given by Professor Dr. Sathaporn Chitta Palapong, the Dean of Veterinary Faculty of Veterinary Technology, Kasetsart University. And we will start the conference very soon.
Good morning. On behalf of the Faculty of Seminar, Academic Veterinary of Australia, scientific knowledge. So as I wish this good morning or good morning.
For those of you who have not registered to the conference, please do so as the conference will begin shortly. The registration form was posted in our website. The conference is going to start very soon. While we are waiting for the conference to start, please allow me to give you a brief detail for today scientific program. This is the third Veterinary Technology and Nursing International Seminar, Engaging Science for Animal Health Innovation, organized by Faculty of Veterinary Technology, Kasetsart University, Thailand. This conference is held on 15 and 16 December 2021. Today is the theme of animal health and environment. We have 11 speakers for this meeting. In the first morning session, we will have a warm welcome address by the Dean of Faculty of Veterinary Technology, Professor Dr. Sthaporn Chitta Palapong. Then we will have the first talk from Associate Professor Dr. Hiroshi Shimoda from Laboratory of Veterinary Microbiology, Yamaguchi University, Japan. And at the end of the day, the conclusion will be given by Professor Dr. Sthaporn Chitta Palapong, the Dean of Faculty of Veterinary Technology. ฮัลโหลต้องได้ไหมครับพี่หนึ่งฮัลโหลได้ยินนะครับ
ได้ยินค่ะ
Good morning, the Dean of Faculty of Veterinary Technology, honorable speakers, distinguished guests, ladies and gentlemen. On behalf of Faculty of Veterinary Technology, I would like to extend a very warm welcome to all of you to the third Veterinary Technology and Nursing International Seminar, Engaging Science for Animal Health Innovation, organized by Faculty of Veterinary Technology, เกษตรศาสตร์ University, Thailand. Let me first begin by introducing myself. I am Associate Professor Dr. Chainarong Skunthaw. I will be your MC today. To kick off this morning session, I am pleased to invite Professor Dr. Sthapon Chitta Palapong, the Dean of Faculty of Veterinary Technology, เกษตรศาสตร์ University, to give a warm welcome to all of you who those joins the conference. Please give a big round of applause for Professor Dr. Sthapon Chitta Palapong.
Good morning. On behalf of the Faculty of Veterinary Technology, เกษตรศาสตร์ University as the host of this special seminar, I would like to welcome all distinguished delegates, researchers, academic staff, and participants to the third Veterinary Technology and Nursing International Seminar in the topic of the engaging science for animal health innovation. I really appreciate all speaker contribution in this seminar and would like to thank for your excellent presentation and devoted time for this seminar. The topic of animal health and environment is leading to several kinds of knowledge for human and animal health as well as the environment that might be beneficial for all participants. Finally, I would like to thank the organizing committee, honorable speaker, participants, and sponsors for strongly support this seminar and wish this seminar have the successful outcome. Thank you.
and he obtained his PhD from Department of Veterinary Microbiology, the United Graduate School of Veterinary Science, Yamaguchi University, Japan, and his main research target in surveillance of wild animal viral diseases, arthropod-borne viral disease, and exploring novel virus from various animals. Ladies and gentlemen, right now I will give you for the Associate Professor Hiroshi Shimoda for the speak for today. Please go ahead.
Can you see the slide? Okay. Okay.
Yep.
Okay. Thank you for introducing me. I'm Hiroshi Shimoda from Yamaguchi University, Japan. Today, I would like to talk about exploring novel zoonotic viral diseases. So it leads to the prediction and prevention of future pandemics. First, I would introduce the One World, One Health. All of you probably know about this, but it means that sharing of responsibilities and coordinating global activities to address health risks at the animal-human ecosystem interface, and preventing animal and public health risks attributed to zoonosis and animal diseases impacting food security. And based on that, many unknown viruses are there in the world. And also no information on their pathogenicity and transmission cycle and their route. And zoonotic viral diseases is a significant threat to public health and livestock industries. And internationalization and climate change may alter its distribution. So we have been conducting surveillance among various animal species. So today's topic is that it's all actually ongoing studies, but one is comprehensive surveillance of viruses among hedgehogs. So detection of adenovirus and herpes virus, and also isolation of hedgehog adenovirus from meerkat, and characterization of hedgehog adenovirus. And the other one is surveillance of bat-derived viruses in Japan. I will talk about detection of the adenovirus and herpes virus. So first, I would like to talk about comprehensive surveillance of virus among hedgehogs. The background is that hedgehogs are getting popular as a pet in Japan, and many of it is actually imported from Thailand. And the one species is that African pygmy hedgehog is the only species that can be raised as a pet in Japan. And as increasing the population of hedgehogs in Japan, there are many hedgehogs visiting animal clinics with various clinical signs. But the reason, the background of these diseases are not known. And one example is that there is detection of adenovirus from lung of fatal cases of respiratory symptoms. So we decided to explore viral infectious diseases among captive hedgehogs and comprehensive surveillance of herpes virus and adenovirus among hedgehogs visited animal clinics. So these are the material and methods. We collected 150 swabs from hedgehogs, and we used PCR using adenovirus consensus and also herpes virus consensus primers. resulting some positives from both adenovirus and herpes virus. And this is a result of sequencing of detected adenovirus. Actually, we detected the same virus that was isolated from the fatal case of pneumonia in hedgehog. So it was 100% homology with African pygmy hedgehog adenovirus one. And for herpes virus, actually, we detected a novel beta herpes virus. So it's actually closely related to through cytomegalovirus, but it's only 56% homology in amino acid. So I think it's a novel herpes virus. And we actually detected viral genomes, those viral genomes using a specific primers, and resulting 3 out of 150 are positive in adenovirus, and 17 out of 150 are positive in herpes virus. And correlation, this table shows the correlation between clinical signs and infection. This is for herpes virus. So there's a significant difference in neurological disease hedgehogs that there's more positives in with the neurological disease hedgehog. And interestingly, the 49 hedgehogs which had neoplastic disorder, so tumors, were not novel herpes virus positive. So this herpes virus may be affect the development of neoplastic disorder. We need further examination on that. And for African pygmy hedgehogs, the number of the positives are not so high. So even if there is a significant difference, we cannot say much about this. But for respiratory disorders, there is a significantly higher. And also some age, so younger age hedgehogs actually showed higher positive rate. So for so far, a brief summary, we could detect novel herpes virus and hedgehog adenovirus from hedgehogs. And there's correlation of novel herpes virus and clinical signs may relate with neurological diseases or may affect tumor development. We need further examination on that. And for hedgehog adenovirus, some of them may be subclinical infection, even though this virus actually was isolated from fatal cases of hedgehogs. Some of them may be subclinical infection. And infect soon after their birth, because the young hedgehogs showed higher positive, and eliminated as they grow, maybe. So actually, this virus, African pygmy hedgehog adenovirus, is very interesting virus. This virus is closely related to skunk adenovirus. And skunk adenovirus is actually was isolated first from wild skunks in Canada with acute hepatitis and pulmonary edema. And it is closely related to canine and bat adenovirus. In addition, the captive marmosets, so the primates with severe respiratory symptoms, showed also the same virus. Also in captive gray fox with pneumonia in USA, wild porcupine with pneumonia in USA, and hedgehog with respiratory symptoms in USA. And in Japan, also detecting from African pygmy hedgehogs as African pygmy hedgehog adenovirus 1. So these virus infect the various species of animals. Normally, the host specificity of adenovirus is really high. So human adenovirus only infect the human, or canine adenovirus only infect the dogs, and porcine bovine adenovirus only infect the cattle or maybe to deer. So it is very rare case that many animal species can be infected with same adenovirus. So actually we have another data about this virus. We found some meerkat with bronchial pneumonia, so necrotizing bronchial pneumonia, and with inclusion body. And we actually sampled some organs and using adenovirus consensus primers. And also we performed virus isolation. And resulting, we successfully isolated the African pygmy hedgehog adenovirus 1 from this meerkat. And resulting 100% match to that virus. So this virus also infects the meerkat. And further examination in the same facility that meerkat was raised, other meerkats actually was positive against adenovirus, and also bearcat, the other animal species, also showed the PCR positive. So we also identified that many species of animals can be infected with this adenovirus. So using those isolated virus, actually we performed the we examined the growth kinetics of this virus, adenovirus, and resulting propagate well in Vero, which is derived from monkey, and MZCK, which is derived from canine, and CRFK, and also from BHK, which is derived from hamster. So various cell lines can support the propagation of this virus. Also, for further examination of the hedgehog virus, we actually established the hedgehog cell line, and we established two kinds of cell line, and using those cell, one is derived from a granule cell tumor in ovary, and one is from histological sarcoma in right forefoot. And resulting that, using this cell line, we actually tried to propagate those adenoviruses. And resulting, both cell lines are sensitive against African pygmy hedgehog adenovirus 1. So this cell line also can support the virus propagation. Also, we have performed some surveillance of this virus among various animals, so including hedgehogs, rabbit, nutria, deer, wild boar, badger, bear, and resulting that hedgehog showed approximately a half of hedgehogs have been infected by this virus, and many deer and wild boars were positive against adenovirus hedgehog adenovirus. But this actually may be a cross reaction of another adenovirus. So rabbits or nutria or other animals, we could not find any positive so far. So we need further examination on these virus surveillance. So brief summary for so far. We could isolate the adenovirus from meerkats with a respiratory disease. So this virus actually showed symptoms to other animal species. And also we could detect from the other bear cats. And many captive hedgehogs are infected with African pygmy hedgehog adenovirus 1. So we need further examination of its transmission cycle and role. And establishment of 2 strains of hedgehog-derived cell culture, and it will be the powerful tool for analyzing hedgehog-derived viruses. So further research on adenovirus hedgehog, African pygmy hedgehog adenovirus for its possibility as a zoonotic virus is required. So my hypothesis, my research strategy in the future will be that African pygmy hedgehog adenovirus 1 is actually can infect to various species of animals. So we need to identify why this virus can infect to the many species of animals, while human or porcine or canine bovine adenovirus can only infect those animals. So and also we need to find out, is there any spillover to the other animals of this virus? So we need further examination of transmission cycle or pathogenicity or so on. So for prediction of future pandemic. So next topic is that surveillance of bat-derived virus in Japan. So there's many bat-related viruses distributed in the world. So many of the emerging infectious diseases are bat-derived viruses. I will introduce you the bat-derived severe viral infectious diseases. For example, there's rabies distributed in worldwide, and in in Thailand, there's rabies. Also, Henipa virus infectious disease, which is distributed Australia and Southeast Asia. Actually, so far, there's no patient in Thailand. Or Ebola hemorrhagic fever virus is distributed in Central Africa. And SARS, MERS, and COVID-19 is also actually distributed worldwide. And also, hantavirus hemorrhagic fever or hantavirus pulmonary syndrome. It mainly distributed in Eurasia, America. And we have been working on bat viruses in Asian countries, including Thailand, with my previous professor. So sampling and also support and surveillance. And but in Japan, we haven't been actually surveyed much about the bats, especially in Yamaguchi Prefecture. There is a biggest limestone cave in Japan, which is called Akiyoshido. If some of you may have come to Yamaguchi, maybe you might have gone there. And Akiyoshido is a natural monument of Japan, and it's very beautiful. And actually, this cave, in this cave, there's 6 species of bats live there. And but since the Akiyoshido is a national monument, so we cannot sample there. So we actually collected near the Akiyoshido. There is a cave and also the tunnel in Yamaguchi Prefecture. We collected 75 bats in Yamaguchi with 4 species, Minapteras species, and also 2 species of Rhinophilus, and also 1 species of Myotis. And we sampled organs or oral and rectal swabs, and also the serum. These are the bats we collected. And after we enter the cave in Yamaguchi like this, and we find some Rhinophilus bats, and also in tunnel we find like this. And as you know, bats may be the natural host of the COVID or other severe pathogens. So when we collect the samples from bats, we actually wear the personal protection equipment like this, and then we collect bats like that. And as a result of virus detection of herpes virus and adenovirus, actually we could detect a novel adenovirus from Rhinophilus phrenicinum in Yamaguchi, which is actually divergent from the other Japanese isolate. So we need further examination on that. Also, interestingly, for herpes virus, we could detect more than 10 different herpes virus from the bats collected in this study. As you can see, these colors these colors actually means that different species of the bats. And as you can see, the same species actually make the same clusters, but yet you can see YV31 oral here, YV31 brain here. So in the same species of in the same bats, there's several kind of herpes virus infected. So some bats are co-infected with different type of herpes virus. And also one Rhinophilus gamma herpes virus have been isolated, this one, by ferrocell. So also we need further examination on that. I mean, for bat diseases or possibility of those viruses to be a zoonotic or the infect to the other animal. So finally, I would like to show you my original cell culture collection. I have been establishing cell line for cicadea or albor, hedgehog, lion, black-backed tiger, marten, and Nutria meerkat, and so on. So there's many sources of cell lines, so it is welcome to use my cell lines as a collaboration research. Thank you for your attention, and also I would like to thank all for the cooperation against these researches. Thank you.
So Professor Shimoda talking about his group research, which has been detecting and isolating various viruses, including adenovirus, Nipah virus from wildlife, such as skunk, hedgehog, bat, also found in Japan. So from these presentations, if there are any questions from the floor, or you can ask yourself, you can send message to our chat box. Okay, the first questions from box. Why researcher interest to study adenovirus in wildlife? And then, what is virus important in wildlife animal? Please, Professor Shimoda.
I'm sorry. I got the first question, but I didn't get the second question.
Okay, the first question is why you interest to study in wildlife?
Adenovirus. You mean the adenovirus? Yeah. Actually, I've started. First, I was asked to survey. I mean, diagnose the virus infection in hedgehogs and also meerkats, and find out that they have same virus infected. So it means that it can be this virus can be infected to many species of animals. So I'm interested in that adenovirus, and I started to work on adenovirus among wildlife. Is that okay?
Okay. And then,
Yeah.
what is virus important in wildlife animal, or these wildlife are reservoir from zoonotic disease in human population?
So you mean what virus is important
Okay.
for
And
what?
okay. Because it's the zoonotic disease also fall in how important to okay from Dr. Supansa. Okay.
Hi, it's my question. Sorry for
Thank
the
you. Thank you.
confusing questions. I wondering, the first question is why you interest to study the adenovirus in the wildlife animal like the hedgehog and the meerkat. And my question number 2 is why the adenovirus is important in the wildlife animals. I mean, in the health, or this virus is a animal. The wildlife animal is a reservoir for the zoonotic disease in the human population.
Okay. I understand your question. Actually, this I'm not sure about these adenovirus. I mean, especially the bat adenovirus for this disease to the bats or not. But we need a further examination on that pathogenicity. And but so as I showed you, the African pygmy hedgehog adenovirus is actually can infect to various species of animals, and also can cause the especially on the respiratory syndrome. So and also in primates. So in marmosets, they found same virus. So this virus may be the candidate. I mean, not this virus itself, maybe, but like the virus like this virus may be the candidate of the future pandemic among humans, maybe. So that's why I'm interested in these virus studies. Is that okay?
Thank you for question and the last question. I can reach up your slide in the first. So I'm wondering how close the genetic relationship among the adenovirus in the wildlife and the human are. Do belong to the same clade or the totally belong to the different clade?
For human adenovirus and the adenovirus that I detected is totally different, but same family, mast adenovirus, not family, same genus, mast adenovirus, the same genus, but totally different cluster. So probably they have a different character.
Thank you for your question. Can I ask for more questions? I just wondering the prevalent rates of the adenovirus that you study. Did you study about the prevalent or the incident rate?
What
prevalent
is it?
or incident? Prevalent or
Yeah.
incident. Did you study about that? For the
Did I study? I'm sorry. I'm sorry, I didn't copy. Please say that again.
I just wondering about the prevalent and incident
Prevalence. Yeah.
of the adenovirus that you study in the wildlife animal. Did you study that?
Yeah. You mean the prevalence? Yes. Actually, it's now ongoing, and I surveyed among wild boar or deer, and they had they showed a high prevalence. But I think it's not that they are infected with African pygmy hedgehog adenovirus. Probably it's they're infected with other adenovirus closely related to those adenovirus. So it should be a cross reaction. We need further examination on that. And we will actually try to survey more other species of animals.
Thank you so much for the wonderful question. Thank you.
Pansa and also Professor Shimoda has any questions from the floor.
Yep.
Okay, he has no questions. I will move to the next speakers.
Okay.
Thank you very much for Professor Shimoda. Thank you.
Thank you very much.
Thank you so much. For the next speaker is Assistant Professor Dr. Chumpai Terakawa from Laboratory of Comparative Toxicology, Department of Animal Science and Biotechnology, School of Veterinary Medicine, Asabu University, Japan. And let me give for his background. He earned PhD from the United Graduate School of Veterinary Science, Yamaguchi University in 2009. And he also works in Division of Reproductive Science at Cincinnati Children's Hospital Medical Center, Ohio, United States of America. And right now, he is an assistant professor in Asabu University. His research focusing on female reproductive biology, including the development of female reproductive tract, mechanisms for the establishment and maintenance of pregnancy, and the etiology of gynecologic cancer. Also, he works on genome editing in embryos to create genetically modified animals. Ladies and gentlemen, I give you for Associate Assistant Professor Chumpai Terakawa. Please go ahead.
Hi, good morning, everyone. Can you hear me?
Yes.
Okay, thank you. So first of all, I'd like to thank you for the coordinator to giving me such great opportunity. So this is Junpei Terakawa from Asabu University in Japan. And today I'm going to talk about one of my research project about female reproductive biology. The title is Cell-Fate Commitment of the Mammalian Duct Epithelium. So when I saw the schedule of this meeting, my topic is quite different from others. So I hope you have some interest in this field. So today's my topic is about female reproductive tract, especially epithelial differentiation of mammalian duct. So first, what is mammalian duct? So in mammals, the majority of female reproductive tract arise from a pair of mammalian ducts. So the name is come from the German physiologist who first described in this duct. So during development, both male and female has two ducts called Wolffian duct and Mullerian duct. So Wolffian duct becomes male reproductive tract. On the other hand, Mullerian duct give rise to female reproductive tract. So Mullerian duct comes from the both sides it's the beginning is the invagination of ceramic epithelium, and the mammalian duct elongated and then fused in the center and then expanded to the urogenital sinus. So lower part become vagina, and then cervix, and the center part become uterus, and then continue to the fallopian tube or oviduct. So in human case, from upper third, 1/3 of vagina to uterus and then fallopian tube, these all come from the mammalian duct. And the mammalian duct is undifferentiated, very simple duct. So as you know, the female reproductive tract has different function, different feature in their region. So the uterus can support pregnancy. So uterine epithelium is simple columnar, and they have ability to secrete and form glands. And on the other hand, ectocervix or vagina, I call it vaginal epithelium here. So the vaginal epithelium is stratified and squamous, and they are keratinized. So why do we need to know the mechanism of epithelial differentiation? It goes back to the 1940s. So the health problem induced by DES, diethylstilbestrol, has been reported. So DES is an orally active non-steroidal estrogen, first synthesized in 1938. So DES has a similar effect of 70-beta estradiol. So DES has been used by millions of pregnant women to prevent miscarriage from the 1940s to 1970s. But however, DES has no positive effect to prevent miscarriage. On the contrary, DES daughter who expose the DES in utero frequently have developed the disorders of cervix and uterus. From that time, the molecular mechanism for action of DES has been investigated. So the health problem induced by DES exposure is the vaginal adenosis. The definition is the development of simple columnar epithelium in the vagina. So that is, the vaginal epithelium should be stratified, but the simple columnar epithelium is there. So as high as 90% of women who are exposed to DES in utero develop vaginal adenosis. So vaginal adenosis itself is a benign tumor, but it is considered to become cancer, vaginal adenocarcinoma. And then the risk in DS daughter is 40 times higher than compared to the non-DS daughter. And vaginal fornix here is the most sensitive to DS treatment. So from 1970s, the study using mouse model has been established to know the mechanism of epithelial differentiation. Here, the study of tissue recombination and performed by Dr. Cunha. He collected the uterus and the vagina from newborn mice. And he separated the epithelium and mesenchyme by enzyme treatment. Then he reconstructed these tissues. So uterine mesenchyme plus uterine epithelium, or vaginal mesenchyme plus vaginal epithelium. That's a normal combination. So he mixed uterine mesenchyme plus vaginal epithelium, or vaginal mesenchyme plus uterine epithelium. So these mixed tissues were implanted on the kidney capsule of the other mice, then grown up for 1 month. So the result is, so normal condition, normal combination, the uterine epithelium becomes simple columnar on the other hand, vaginal epithelium becomes stratified. But if mixed these combination, if vaginal epithelium is combined with vaginal mesenchyme, this epithelium becomes simple columnar. If uterine epithelium was combined with vaginal mesenchyme, the uterine epithelium becomes stratified. That means the organ-specific mesenchyme, that is uterine or vaginal. So this mesenchyme determines epithelial sulfate. And the sulfate of Müllerian duct epithelium established during first 5 days of postnatal life in mice. So the mice is good model to study to know the mechanism of epithelial differentiation. So when we give the DES in the neonatal mice soon after birth, we can induce the vaginal adenosis. And then continuous study molecular mechanism, the key transcription factor delta NP63. So this transcription factor determines the epithelial sulfate. So here's a model of the DS treatment of the mice. So as you can see, the P63, delta NP63, the transcription factor should be expressed like this brown color. But when we give the DS, so the expression was disrupted. So the P63 is a homologue of the tumor suppressor P53, and it's highly expressed in the basal or progenital layer of many epithelial cells. So P63 has two dominant isoforms. One is TA form, and the other one is delta N form. They have truncated N-terminal. So as you can see here, so the stratified epithelium in the cervix, the vagina or skin, they all express delta N isoform. So delta NP63 is a dominant isoform expressed in the squamous epithelium and then kill a regulatory factor. So Müllerian duct epithelium is undifferentiated epithelium, and then it has simple columnar. When the signal from the vaginal mesenchyme comes in, the P60 expression was induced. So once delta and P63 expression was induced, this undifferentiated epithelial cell can differentiate into the vaginal epithelium. So they can make a stratified. So, and on the other hand, delta NP60 negative cell can differentiate it into uterine epithelium, and which is simple columnar. So when I joined this project, the question was, what a signal from the mesenchyme, and what a target of DES. So here's a model of vaginal epithelial differentiation by 2060. So our team reported that 3 independent signal pathway is essential to induce delta NP63 in the epithelium. So BMP4 and activin A and FGF17. So actually the not 3 and 4 factor. So these are stromal factor. So these stromal factor can bind to their receptor expressed in the epithelium and they activate downstream target. So BMP4 activates SMART and then activin A activated RANKS1 as a transcription factor, and then FGF7 and 10 can bind to their specific receptor FGFR2/3B and then activate MAP kinase. So all 3 signaling pathways are essential for expression of delta NP63. So to know these pathways, we use a lot of genetically modified mouse, especially tissue-specific gene knockout. So I'd like to explain the system briefly. So we use the conditional knockout by Curie-Loxby system. To use Curie-Loxby system, we need to prepare two different mouse line. One is Curie transgenic line, which is that mouse carrying the Curie recombinase gene controlled by tissue or cell-specific promoter. And the other one is the mice called Flox mice. So the certain sequence, the target gene is sandwiched by the certain sequence. So when Curie recombinase expressed under the specific promoter, this Curie recombinase can recognize the certain sequence and then can eliminate the gene. So we crossed Curie transgenic line and then Flox mouse, and we need several crosses. And then finally got mice carrying the Curie expressing gene and then target and Flox allele of target gene, and then we can make a conditional gene knockout. It might be complicated. So in our case, in most our case, we use the Wnt7A Curie line. So Wnt7A is activated only in the Müllerian duct epithelium. So we can make a Müllerian duct specific gene knockout. We cross the Wnt7 Curie with our target Flox mice of our target gene. Then when the gene X is Flox and wild type, this mouse would be a conditional heterozygous of this target gene. So we use these mice as a control. So when the gene X has the Flox, this gene was eliminated completely. So we use these mice as the conditional knockout. So we knew the three independent signal pathways is essential to induce delta NP6C expression, but we don't know how these three different pathways regulate P63 expression. So we checked our gene expression profile and then found another transcription factor named 6-1. So here, the 6-1 is enriched in the vaginal tissue. So 6 gene family have 6 different, it's also complicated, 6 different gene, and then 6-1 play important roles in ear, kidney, and skeletal muscle development. So first, we check the 6-1 expression in the vaginal tissues. So here the result of Western blot. As you can see, 6-1 is only expressed in the vaginal region, not in the uterus. So LANX1 is also essential factor, essential transcription factor. It's also expressed only in the vagina. So here the result of immunofluorescence. So P63, these areas should be stratified, but P63 is not expressed in the postnatal neonatal day 1, but these essential transcription factor is expressed. So we found that 6-1 expression is followed by the LANX1. So here the border of stratified epithelium in the cervix to vagina or simple columnar epithelium in the uterus. Here's the border. So as you can see, the nice expression of LANX1 and then 6-1 expression followed by LANX1. And then next we check the 6-1 expression in the different kind of various gene knockout model. So SMART4 is also essential. When we knock out SMART4 in the Müllerian duct epithelium, so there's no P63 expression like this. So this is control. So you can see the red signal. And then LANX1 expression is still in the SMART4 knockout. However, 6-1 expression is completely lost in SMART4 conditional knockout. That means 6-1 is downstream of SMART4. So next one is the LANX1. So LANX1 knockout, this LANX1 is also essential factor. So when we knocked out LANX1, the P60 expression is completely disrupted, and then no LANX1 expression. So 6-1 expression was decreased in LANX1 knockout. That means 6-1 and LANX1 have some collaboration. And then the third factor, FGFR2, that's a receptor. So we make a receptor conditional knockout. So this receptor and then MAP kinase activation is essential. So when we knocked out FGFR2, the P60 expression, delta P60 expression is completely lost. But 6-1 expression is still there. That means 6-1 expression was not affected by the FGFR2 duration. So it's not related to this pathway. Then next, we make a 6-1 conditional knockout in the same using the same strategy. So unfortunately, we only have a very minor effect of P60 expression, as you can see here. So very small part of the vaginal fornix lost the P60 expression. So we combined 6-1 mutation with LANX1 mutation, and then found that they have gene dose dependent regulation. So here, LANX1 conditional knockout, so it itself has no effect of P63 expression, but if LANX1 conditional head combined with 6-1 conditional head or conditional knockout, the phenotype becomes severe. So that means 6-1 and LANX1 regulate P63 expression in gene dose dependent manner. And finally, so we check the this expression we check the gene expression after DS treatment, and then we found that. So here's a control. So we can see the nice expression of LANX1 or 6-1. So when give the DS in neonatal mice, so LANX1 expression was decreased as well as 6-1 expression was decreased. And then we confirm the expression by a quantitative PCR. So this result indicates that DS was target not single molecule. So DS downregulate both LANX1 and 6-1. So actually, I skipped a lot of data, but here's a current model of vaginal epithelial differentiation by 2020. So in normal development, the 3 independent signal pathway is activated from the mesenchyme. So activin A, BMP4, FGF7, and 10. So these molecules bind to the receptor and then activate their downstream. And then we found the other essential factor, 6-1, is collaborated with this signaling pathway and then regulate p63 expression. So once p63 expression was activated and the delta and p63 is expressed in this undifferentiated epithelium, itself can promote its expression, and then this undifferentiated epithelial cell have a vaginal fate, and they can stratified. So during development, if the DS was exposed, DS can bind the estrogen receptor and then downregulated RANKS1 and 6-1. So this causes the disruption of this signaling pathway, and p63 is never expressed in this undifferentiated cell, so it has a uterine fate. So that's why DS induced vaginal adenosis. So here's the acknowledgement of these topics, topics of my research. Thank you for your attention. That's all for today.
So thank you very much for Dr. Jubei Tarekawa. He talking about the Mullerian duct, which is the part of female reproductive tract, and it is committed to become stratified squamous epithelium of the vagina and ectocervix. He investigated the vagina epithelium by determining the transcription factors such as NP63 expressions. And also, he also construct the engineer mouse by using Krelok system and study the expressions and factors that involve in the expressions. So, firstly, may I ask for any question from the floor or from chat box? Okay, maybe the first question from me. So because you use DES for study the expressions of the transcription factors, why this study you use DES to study the repressions of transcription factor? And also you recommend that not only DES, there are some estrogenic compounds that can repress and induce the factor. Could you please explain
Yes.
more?
So why I use the DS is DS is used in the pregnant woman in 1940s. So it's actually estrogenic compound used in the human.
Okay.
And, so that's why we focused on the DS itself. And as you said, the other estrogen compound also have this similar effect or same effect.
Okay. And how about the other estrogenic compounds? They also
also had the same effect when we give the when we experimentally give the mice.
Okay, and not only for determining the transcription factors, for the other factors or like protein levels, will you determine that?
We do not actual expression of the transcript factor when we give the other compound, but
Okay.
it's actually difficult. The other compound also has an estrogenic effect, but the activity or how to say the ability to bind the receptor is different from the compound. So
Okay.
when we give more, we can make a same. We can make a we can see the same effect, but it's different to set how volume we could give.
Okay. So thank you very much. Your presentation is very nice and interesting for me.
I think it's very difficult. So and different field. So thank you very much.
Yeah. So thank you very much. May I move to the next speaker?
Thank you, Assistant Professor Dr. Chumpai Terakawa from Asabu University, Japan. And the third speaker on this session morning is Dr. Vanas Sicharan from Faculty of Veterinary Technology, Kasetsat University. Please let me give his background a little bit. He earned Doctor of Veterinary Medicine from Kasetsat University at 2005 and master degree in agricultural biotechnology from Kasetsat University in 2016, and he earned Doctor of Philosophy from agricultural biotechnology from Kasetsat University in 2021. And his research interests molecular epidemiology of zoonotic parasites and parasitic infections of non-human primates. Ladies and gentlemen, I give you Dr. Vanat Sicharan, please go ahead.
Sawasdee krab, and good morning, everyone. Thank you for the MC for introducing me. I am Vanat Sicharan. I'm the staff at the Faculty of Veterinary Technology, Kasetsat University. Today, my topic is about potentially zoonotic parasites among the long-tailed macaques or Macaca fascicularis in Thailand. The first, I would like to talk about the macaques in Thailand. In Thailand, there are many species of monkey and macaques. And the most observed species of macaques in Thailand is long-tailed macaques, or the scientific name is Macaca fascicularis. This macaque may be called crab-eating macaques or cynomolgus macaques in the laboratory. In Thailand, we can find these macaques in almost every region in Thailand, except in the north of Thailand. The red dots in this picture show the area that these macaques live in the Thailand. And if you used to travel in Thailand, or maybe when the situation of COVID-19 is okay, if you come to Thailand and you travel in Thailand, you may be found this macaque in many tourist attractions, such as in the San Prakan Chai and Prapang Sam Yot Temple in Lopburi Province in the central of Thailand, in Prachop Khiri Province, in the Phuket, in the south of Thailand, in Songkhla, in the south of Thailand, in Chonburi, in the east of Thailand, and in the Mukdahan province. Many areas that you can find these macaques in Thailand. And these macaques live very close to human community, and they share the environment with the people, with the local residents and tourists, and also the pets. And they can carry the pathogens that are potentially zoonotic to human. So that's why I and my team interest to study the occurrence and the genetic diversity of the zoonotic parasite in the long-tailed macaques in Thailand. The information in this presentation come from my study about during 4 to 5 years ago. In 2014, we collect 200 fecal samples from the long-tailed macaques in the Lopburi province at the central of Thailand. This area is the very famous tourist attraction in Lopburi province. We call San Prakan Chai and Prapang Sam Yod temples. And we collect the feces from the long-tailed macaques in this area. And during 2016 to 2018, we collect the blood sample from the long-tailed macaques in 9 provinces of Thailand. This is the location that we collect the blood sample from the long-tailed macaques. In the central of Thailand, we collect from Lopburi and Samut Songkhram province. In the west, we collect in Prachuap Khiri Khan province. In the south, we collect in Phuket, Phathalung, and Songkhla province. In the east, we collect in Chonburi province, and in the northeast, we collect in Mukdahan and Amnacharan province. The total of the blood sample was 715 samples and composed of male macaques 470 sample and female macaques 245 sample. And then all of the fecal samples and blood sample were taken for DNA extraction. And then the extracted DNA were used to test by molecular analysis. And this is the zoonotic parasite that we found in our study. The first parasite that we found is the protozoa called Giardia. We found in 14 samples from 200 samples. And we use the method to test for Giardia is based on nested PCR. And from the sequence analysis, we found that the Giardia in this study was Giardia duodenalis. And when we classify into the assemblage level, we found that most of them was the assemblage B. Only one sample was the Giardia duodenalis assemblage A. And one sample that cannot identify. This is important because for the human giardiasis, only Giardia duodenalis assemblage A and assemblage B that can cause the human giardiasis. And then when we found in the long-tailed macaques, it means that the long-tailed macaques may be act as a reservoir host for this parasite to the humans. And the transmission of these protozoa is very easy because they can transmit via direct contact, via fecal oral route. So in this picture that we saw that the human very close contact to the long-tail macaques that have the chance that this protozoa can transmit to the human. The next parasite was Cryptosporidium. We found 1% to sample from 200 sample of fecal sample that are positive for Cryptosporidium species. And for the sequence analysis, we found that the Cryptosporidium was classified as the Cryptosporidium parvum. And then we used the PCR RFLP procedure to classify the genotype of Cryptosporidium parvum that we found. And we found that Cryptosporidium parvum that we found in our study were classified as the monkey genotype. The next parasite is the helminth in the intestinal tract of human and non-human primates. It is the ticulid or whipworm. We found the ticulid in 3 samples from 200 samples. And we use the nested PCR to detect the ticulid. And the sequence of ticulid that obtained from our study share 98 to 99% identity with the sequence of ticulid trachea from human in Thailand. And from the phylogenetic tree based on the ITS1 show that sequence of the ticulid in our study that show in the triangle mark, they were classified in the same crate with the ticulid trachea from human and ticulid trachea from another non-human primate in many countries. And the next parasite was Hymenolepis. That we found this parasite 8 sample from 200 fecal sample. By using the conventional PCR, the sequence that we found share 97 to 100% identity with the sequence of Hymenolepis diminuta. And from the phylogenetic tree based on the partial 18S rRNA gene, the sequence that obtained from our study were classified in the same crate with the Hymenolepis diminuta from the bile rat in other countries. Okay, so that was the parasite that we found in the fecal sample. And then when we detect the hemoparasite from the blood sample of the long-tail macaque, we found 1 sample that positive for Bartonella in the Chonburi province. And we have the positive sample that positive for hemoplasma in many provinces in Thailand. We test for the Plasmodium and Babesia as well, but in our study, we cannot find the positive sample for Plasmodium and Babesia. For the Bartonella, we detect only 1 sample from the male macaques from Chonburi province based on 16S rRNA gene, GLTA gene, and FTSZ gene. that give the same result that the Bartonella that we found was the Bartonella quintana. So because we found only one positive sample, so we cannot find the association between Bartonella infection and location or sex of the macaques. Bartonella quintana that we found show very high identity with the Bartonella quintana that previously reported in the gene bank. For 16S RNA gene, they show 100% identity with the Bartonella quintana from Japanese macaques and rhesus macaques in the previous study. And GLT8 gene and FTSZ gene as well show the about 99% identity with the Bartonella quintana from Japanese macaque and rhesus macaques. And for the genetic analysis, we perform the phylogenetic analysis by using maximum likelihood methods based on 3 genes, 16S, rRNA gene, and GLT8 gene, and FTSZ gene. And the results is the same, that Bartonella quintana in our study was classified in the same grade with the Bartonella quintana in rhesus macaque, Japanese macaques in other country. And then we perform the phylogenetic tree of Bartonella based on the combined gene, combined of 16S, RNA gene, GLT8 gene, and FTSZ gene by using the Bayesian method, and the result is the same. For the Bartonella quintana is the gram-negative intracellular bacteria, and these bacteria can infect the human. The vector of this bacteria is the human body louse and can cause a broad spectrum of disease, such as train fever, endocarditis, myocarditis. So in our study, we can find a low prevalence of Bartonella quintana in long-tail macaques from Sumeri province. However, it is the natural infection of Bartonella quintana in the long-tail macaques, and phylogenetic analysis based on these 3 genes confirm that long-tail macaques in Thailand carry Bartonella quintana. Previously, reports of the Bartonella quintana in long-tail macaques have been reported in long-tail macaque in USA and China. And this is, to our best knowledge, is the first report of the Bartonella quintana in macaques in Thailand. And for this reason, this finding suggests that long-tail macaques might serve as a reservoir host for Bartonella quintana. The next hemoparasite is the hemoplasma. We found hemoplasma in many locations in Thailand. The total positive sample was 79 from 715 samples. It was about 11%. And in the male, the prevalence in male was 12%, that higher in the female, that the prevalence was about 8%. And the high the highest prevalence we found in the Mukdahan province, and the lowest prevalence in Prachuap Khiri Khan. There is no positive sample in the Prachuap Khiri Khan province. However, there is no statistically significant association between the hemoplasma infection and the sex or the location of the macaques. The sequence analysis of mycoplasma that we found in this study revealed that the hemoplasma was classified as the candidatus Mycoplasma hemomacaque, that similar to the previous study in the Cynomolgus macaques in the USA and Japanese macaque in Japan. And for the phylogenetic analysis, show that the candidatus Mycoplasma hemomacaque in our study were classified in the same grade with the candidatus Mycoplasma in the previous study from Thailand, from USA, and from Japan. Not only candidatus Mycoplasma hemomacaques that found in the macaque, there were the reports of the other hemoplasma in the non-human primate as well, such as candidatus Mycoplasma aortae, candidatus Mycoplasma carhanii. In human, hemotropic mycoplasma or hemoplasma in human may be the rare case, and almost every case in human suspect to come from the domestic animal, such as come from the cat, pigs, goat, and dog that can found in the patient, can find the hemoplasma of these animals in the patient. For the hemoplasma from macaques, now is don't have the report or evidence that mycoplasma in macaques can transmit to human, but it is important to study in the future that these mycoplasma can transmit to the human or not. In this study, we don't found the significant difference in the prevalence. However, when compared between male and female macaques, we found that the male macaques have the higher prevalence than female macaques. That may be due to the aggressive behavior of male animals. And hemoplasma is the parasite that transmit via the vector. However, they have a limited number of the study on the vector of hemoplasma among non-human primates. So to reduce the spread of hemoplasma in the macaques and also reduce the chance to transmit to other animals, is very important to study about the vector of this parasite in the future. Okay. Conclusion. The result of our study confirmed the natural infection of human pathogenic parasite in long-tail macaques in Thailand, including Giardia duodenalis, assemblage A and B, Trichulis trichiura, Hymenolapis diminuta, and Bartonella quintana. And this study also confirmed the widespread of candidatus Mycoplasma hemomacaque infection among long-tail macaques in Thailand, and also confirmed the natural infection of Cryptosporidium parvum, monkey genotype, in the macaque as well. And the further study of the vector of these pathogens need to be performed to fully understand the transmission cycle between the reservoir animal, arthropod vectors, and human in Thailand. And the last, I would like to thank you, Professor Dr. Sthaphon Chittapalapong, Assistant Professor Dr. Thewin Inpankeo, Assistant Professor Dr. Srawan Keomongkol, Ms. Suphakan Keochod, the veterinarian officer from the Department of National Park, Wildlife and Plants Conservation. And our research was funded by Kasetsart University Research Development Institute. And thank you for your attention. ขอบคุณครับ
Thank you very much for Dr. Wanat Sijalan. So your presentation is very interesting and okay. So now we know that macaques maybe acts as a reservoir host for many zoonotic pathogens which can be transmitted to human. So Dr. Wanat, may I ask you the questions because you found a lot of zoonotic parasites as you mentioned. So which species can be transmitted to human?
Thank you for the question. This the slide what shows. These parasites that can transmit to the human, the protozoa, Giardia duodenalis, assemblage A and B, the helminth, Diculiticula, or whipworm, Hymenopus diminuta, and the bacteria Bartonella quintana. All of these pathogens can transmit to human. These pathogens have the evidence that can transmit to human. However, the other pathogen that found in our study, like Candidatus mycoplasma haemomacaques and Cryptosporidium pavum monkey genotype, although nowadays they don't have the evidence that can transmit to human, but it's like in the future is maybe not sure that these pathogens can be transmitted in the human. So we have to maintain to study about these pathogens.
Thank you very much. So the question from the floor. If participants would like to ask the questions, you can raise your hand through the website. And also you can text message in the chat box. I will deliver your questions to the speaker. Okay, so Dr. Wanat, because there are a lot of parasites that you can identify, and there are some species that can transmit to humans. So, what is the sequence identities between zoonotic that you can isolate from macaques and other wildlife animal? Do you have any idea?
For the sequence identity for other wildlife, in each parasite, they will have the range of hosts. So if we interest in the other wildlife animal, maybe we found the other species, not the same species, but it can many species of the pathogen in wildlife can transmit to human as well. But maybe if we study in the other wildlife, we will not found these species, yes, of the parasite. Yes, but is very interesting as well to study in the other wildlife. Yes.
Thank you very much. So, okay, we move to the next speaker if no one has questions. The next speaker will be Professor Dr. Ryusei Kuwata.
I will give for his background from for Associate Professor Dr. Ryusei Kuwata. He graduated PhD from the United Graduate School of Agriculture, Kagoshima University, Japan in 2007, and he works in Division of Medical Entomology in the National Institute of Infectious Disease, Tokyo, Japan, as first postdoc between 2007 and 2013. During the term, he focused on the study about arthropod-borne viral disease transmitted by mosquito and ticks. He also worked in laboratory of veterinary microbiology under Professor Dr. Maeda, who is one of a famous and leading researcher for veterinary microbiology and zoonotic disease in Japan, in Faculty of Veterinary Medicine, Yamaguchi University. And 2019, he is Associate Professor in the Faculty of Veterinary Medicine, Okayama University of Science, Japan. And his research project focusing on zoonotic viral disease, including arthropod-borne viral disease transmitted by mosquito and tick. Right now, ladies and gentlemen, I give you Associate Professor Dr. Ryusei Kuwata. Please go ahead.
Okay, can you hear me?
Yes.
Okay, thank you. Thank you for your introduction. Hello, everyone. I'm Ryusei Kuwata from Japan. First of all, I would like to say thanks for staff of Kasat University for preparing the symposium and inviting me to speak today. I would like to talk about our recent research for arboviruses. Today, I will talk three topics as below. Firstly, I would like to talk about the brief introduction of arboviruses. Second, I will talk about the genetic characterization of various viruses isolated from mosquitoes in Asian countries. Finally, I would like to talk about the distribution of tick-borne viruses in Japan. In the world, we have been exposed to many kinds of zoonotic diseases, and these viral diseases have been characterized with several transmission routes. For example, rabies was transmitted directly by bite of infected animals. Some of them required another intermediate host for the transmission. Blood-sucking arthropods, such as mosquitoes and ticks, transmit several viral pathogens. These arthropod-borne viruses are called arbovirus. Because many arboviruses are maintained by animals in nature, they are known as causative agent for zoonotic diseases. This table shows a classification of major arboviruses transmitted by mosquitoes and ticks. Flavivirus, alphavirus, virus, also myxovirus, reovirus, rhabdovirus are included arboviruses. Today, I would like to talk about Japanese encephalitis virus, Zika virus, and severe fever with thrombocytopenia syndrome virus. Before I worked in the present position, I worked in Yamaguchi University from 2013 to 2018 under supervisor Professor Ken Maeda. He is one of famous and leading researcher for veterinary microbiology and zoonotic diseases in Japan. Now he moved to Japan National Institute NIID. During the term, we conducted arbovirus surveillance in Asian countries, collaborating with Thailand Katsat University, Philippines Mindanao Central University, and Indonesia Bogor Agricultural University under E-Asia project. Today, I will introduce the partial result of this project. In 4 countries, we collected mosquitoes in residential area and animal houses by using sweeping net, aspirator, CGC dryer trap, and BG sentinel traps. Animal serum were also collected by collaborators and analyzed them. From collected mosquitoes and animal serum, we performed virus isolation by using 2 mammalian cells, Vero and BHK, and 1 mosquito C636 cells. After at least 3 blind passages, we conducted RT-PCR from RNA extraction of culture supernatant. RT-PCR to detect viral genome. We used 5 consensus primer sets for detecting flavivirus, alphavirus, flavivirus, and rhabdoviruses. But for RNA samples, whose supernatants showed clear CPE, but could not detect any viral genome by RT-PCR, we used NGS analysis using Illumina MySeq or ion-torrent PGM system to identify the viral genome. This slide shows the result of mosquito collections. During the project, we collected more than 50,000 mosquitoes, comprising at least 32 mosquito species belonging to the genus Curex, Aedes, Armigères, Ophlerotatus, Anopheles, Mansonia, Polypteroides, Rhizochita, Ursobodia, and Uranotainia from 4 countries. These collected mosquitoes were sorted into pools and used for virus isolation. This slide shows the result of virus isolation. In this study, we successfully isolate Japanese encephalitis virus from 4 countries, Zika virus from Thailand, and many insect-specific viruses. All viral isolates were obtained by using only mosquito C636 cells, and no virus isolates were obtained from Vero and BHK cells, indicating that mosquito C636 cells might be more efficient for an isolation of arboviruses. Japanese encephalitis virus, called JEV, is a positive sense single-stranded RNA virus in the family Flaviviridae. JV is widely distributed in Asia, and they can be divided into 5 genotypes. JV genotype 1 and genotype 3 are the dominant lineage and have been found across the wide area of Asia. JV1 has been further classified into 2 subgenotypes, JV genotype 1A and genotype 1B. JV genotype 2 is the third most common lineage and has been found in Indonesia, Singapore, Malaysia, and Australia. JV genotype 4 and 5 are rare lineages. Only a few strains of these genotypes have been reported from Indonesia, Malaysia, China, and Republic of Korea to date. As a result of virus isolation, we successfully isolated JVs from each 4 countries, supported by the many collaborators in each countries. This figure shows the result of the phylogenetic analysis based on the full ORF genome sequences. This result demonstrated that the Japan isolate belonged to genotype 1A, the Thailand isolate to genotype 1B, the Philippine isolate to genotype 3, and the Indonesia isolate to genotype 4. I would like to explain each isolate by enlarging images in the dotted line. The upper panel indicates the Japan JV isolate. The 5 Japan JV isolates belonging to genotype 1A were obtained from Culex tritanincus mosquitoes captured in Yamaguchi in 2030 and 2060 independently. They were most closely related to each other than other JV strains that were isolated from China and Korea recently, suggesting that they were being maintained in the limited Yamaguchi area of Japan. The lower panel indicates the Thailand JV isolate. The isolate we obtained from Thailand in 2017 belongs to JV genotype 1B and clustered with other JV strains that had been detected in Thailand from 1985 to 2005, Myanmar in 2010, Cambodia in 2014 and 2015, and Singapore in 2014. This result indicates that these JVs have been maintained around the closed area for over 13 years. JV is endemic in the Philippines. However, only 3 JV strains, which had been isolated from 1984 to 1986 from pig belonging to JV3, were only available on the NCBI database for phylogenetic analysis. Surprisingly, we found that our 2 Philippine JV strains clustered with them. Our 2 Indonesia JV isolates, which were obtained from Bali Island, clustered with Indonesia JV strain that had been isolated in the 1980s. These results indicated that JV3, genotype 3 and genotype 4 are still active in each country, and further demonstrated that JV has been maintained in Thailand, Philippines, and Indonesia since at least the 1980s. So in our conclusion, the recent JV isolates in this study are phylogenetically related to the JV strains that were reported in the 1980s in each country, suggesting that they had been maintained in each region for a long time. Some JB strains from China, South Korea, and Japan were spread by an ocean, form the same genotype 1A grade, indicating genetic moving between the countries. However, basically, the JV seems to have been maintained indigenously. In the course of this study, we also isolated many kinds of insect viruses. First, we isolate the Vanna virus. The insect-specific fly virus from Indonesia. Insect fly virus is an assigned genus in the family Flaviridae and only grow on only insect cells. In this study, we successfully isolated the three kinds of insect-specific fly viruses: Aedes flavivirus, Culex flavivirus, and self-fusing agent virus. We isolate the Vanna virus from several mosquito species in Indonesia. Vanna virus is a family Leoviridae, genus Ciadorna-viruses. Vanna virus is first isolated from the cerebrospinal fluid of encephalitis patients in China in 1987. Phylogenetic analysis indicated that our Vanna virus isolates were genetically close to the Jakarta strain, isolated in 1981. We successfully isolated a new alpha tetravirus named Bogor virus from several mosquito species in Indonesia. That virus is an assigned genus of alpha tetravirus in the family Tetraviridie. This virus is also known as insect-specific virus group. Phylogenetic analysis indicated that the virus is a novel virus. We also isolated from the Mesonivirus from several mosquito species in Indonesia. This virus belongs to the genus Alpha Mesonivirus in the family Mesoniviridae. These viruses are also known as insect-specific virus. Phylogenetic analysis indicated that we have successfully isolated two species of Alpha Mesonivirus, Alpha Mesonivirus 2 and Alpha Mesonivirus 3, from Culex trichomicus or Aedes mosquitoes. So in our conclusion, we successfully isolate the variety of viruses only from mosquito C636 cells, indicating that the mosquito cells might be more efficient for an isolation of alpha viruses. Second, we isolated many and various insect-specific viruses, existing the presence of abundant insect virus in mosquito population in Asia. On the other hand, we think the possibility of the competition between alphavirus and insect-specific viruses in the same mosquito pools during the virus isolation, because some researchers suggested that some insect virus suppress the replication of virus inside the mosquito cells or mosquito bodies. So we need further understanding the role of the abundant insect viruses in nature. So next, I would like to talk about Zika virus. Zika virus is a positive single-stranded RNA virus in the genus alpha virus of the family Tocapilidae. Zika virus is found from several kinds of mosquito species, such as Culex, Anopheles, Aedes, Armigerales, and Zika virus distribution is widespread in Asia. Zika virus is an important infectious disease in veterinary field that shows clinical signs in horses and pigs. In horses, Zika virus infection causes transient fever, skin rash, and leg edema. In pigs, Zika virus infection sometimes causes fetal death and reproductive disorder. Recently, it was reported the fever of beef cattle and death of blue fox by the Zika virus infection in China. In Japan, Zika virus combined vaccine is available. For the prevention of Zika virus infection in thoroughbred horses and sows. Recently, we have experienced an Zika virus endemic in Japan. First, from 2014, an outbreak of Zika virus infection had been suddenly occurred among bred race horses and domestic pigs in central Japan. Although they had administered the Zika virus vaccine. Subsequent retrospective study identified the three evidence of the possible origin of the Zika virus outbreak in 2014 in Japan. First, Kobayashi et al. Reported in 2016 that sudden many Zika virus isolates occurred from collected Culex toledanicus mosquitoes in 2012 in summer. Next, we analyzed the serum of wild boars collecting from 2010 to 2016 in Yamaguchi and reported that positive for anti-Zika virus antibody populations had been increased around 2012. Now, we just analyzed the serum of ponies being bred near our university and found the increasing of Zika virus, anti-Zika virus antibodies titer from 2012 to 2017. Phylogenetic analysis of the Japan Zika virus isolate indicated that 2012 mosquito strain and 2014 racehorse strain were genetically different from indigenous Japanese Zika virus strains, but closely related to the strain from China and Korea. These results strongly suggested that the origin of the recent Zika virus outbreaks in Japan were derived from the invasion strain from continental into southwest Japan around 2012, and they were gradually widespread to northern part of Japan by mosquitoes or amplifying wildlife, such as wild boars. In our international project, we investigated the seroprevalence of Zika virus infection among domestic pigs in Thailand from 2017 to 2018. ELISA was used to analyze 1,188 pig serum samples collected from 11 provinces of Thailand during 2017 to 2018, with 23.1% of the sample being positive for anti-Zika antibodies. We found that the positive ratio of anti-Zika antibodies was significantly higher in piglet and all the stages of pigs than in finishing stages, may be caused by the maternal antibody in piglets and many opportunities to be bitten by mosquitoes and acquire the immunity in all stages. Furthermore, we successfully isolated the novel Zika Thailand strain from pig serum and determined the complete genome sequences. Phylogenetic analysis demonstrated that the Thailand isolate was different from recent Zika strains, spreading around the pig population in Japan or China, and formed a cluster with two Zika strains, namely China in 2012 and Far East Russia 1988 strains. So we concluded that two different Zika groups are currently spreading around the pig population in Asian countries. So finally, I would like to talk about the distribution of tick-borne viruses in Japan. This slide shows the images of common hard tick species in Japan. The background is 10-baht coin, which help you to image their size easily. They show in a wide variety of shapes and size, and some of them transmit severe diseases to humans and animals. This is a picture for Amblyomma testudinum showing the different sizes by their stages. You can see that after some days of blood feedings, the ticks becomes several times larger than it was before the blood feedings. So I would like to talk about brief introduction of SFTS virus. SFTS virus is now their taxonomic name on ICTV as Lubei Banda virus. But today, I would like to use the familiar name SFTS virus. SFTS virus is a negative sense 3-segmented RNA virus in the genus Banda virus of the family Phenylepiridae or the Bunyavirales. They are tick-borne emerging virus, and first record was in China in 2012. After that, Korea and Japan in 2013. SFTS is mainly found in China, Korea, and Japan, especially southern part of Japan. This slide shows the SFTS virus cycle in nature. In the field, SFTS virus is basically maintained by ticks and wildlife. There is a tick cycle in which the virus is maintained between developmental stages of ticks vertically, and an animal cycle in which the virus is maintained between virus-infected ticks and blood-sucked wildlife horizontally. During the animal cycle, virus-infected ticks emerged and transmit an SFTS virus to humans. This slide shows the number of case reports of SFTS virus infection in cats and dogs from animal hospitals in Japan until July 2012. The number of pets infected with SFTS virus was much higher in cats than in dogs, and the distribution was concentrated in Western Japan. Currently, some pet owner, veterinarian, and veterinary nurse were infected with SFTS by virus-infected cat, which is being a serious problem in Japan. In 2017, 2 captive teachers in Asa Zoo died by the SFTS infection. At that time, we joined the research team and analyzed the ticks captured around the cheetah's cages. DNA were extracted from the collected ticks and analyzed the blood source animals. We found that partial cheetah genomes from the collected ticks, indicating the indirect evidence that these cheetahs were infected with SFTS by biting virus-positive ticks. This figure shows a positive rate of anti-SFTS virus antibodies in the blood of raccoons collected in some area in Japan from 2007 to 2015. In 2007, no blood sample were positive for anti-SFTS virus antibodies, but from 2008 to 2012, the percentage remained below 10% and gradually increased in 2013 and 2014, reaching at about 50% in 2015. It is thought that the SFTS virus spread rapidly in wildlife during this period in that area. Interestingly, SFTS cases in human and cats had been reported in the same region since 2014, when antibody positive rate began to rise. We think that this may be an evidence that the spread of the virus in wildlife poses the risk of SFTS virus infection to human society. During the surveillance of ticks in Japan, we found several novel viral genomes by RT-PCRs or NGS analysis. From collected mosquitoes and also wildlife serum in Yamaguchi, we found a novel langat-like flavivirus genome for the first time in Japan. Now we start to survey in Oshima Island, where are the lot of ticks and lot of wild boars, and SFTS are also cases in endemic areas. Until now, we detected from collected mosquitoes not only SFTS virus genome, but also novel Kuaranja-like and Kaiseri group viruses. We will conduct further studies to reveal the infection cycle of them and risk of these viruses and prevent them. So this is the final slide. In this presentation, I talked about the spread of alvirus infections transmitted by mosquitoes and ticks. I hope that the data from such epidemiological studies will help us understand the spread of viruses and the route of infection, so that we can prevent the risk of viral infection and contribute to the creation of the safer human societies. So I would like to say thanks a lot for many collaborators, especially to Dr. Ken Maeda and Dr. Amapon for giving me a good experience via international collaboration studies. Thank you very much for your attention.
Okay, Professor Kuwata, do you hear me? Okay, the first question from the box is the Beta virus vaccines for pig nowadays, and which one inactivated, attenuate, or subunit vaccine? Do you hear me? No?
Sorry.
Okay,
sorry.
okay. I hear you already. Okay. So the first question is, nowadays is the Beta virus vaccines for pigs.
Yes, in
Okay.
Japan.
And which one inactivate, attenuate, or subunit vaccine?
Attenuated vaccine.
Okay. And the next question is, what is the age of domestic pigs being vaccinated?
Oh, in Japan, only so mother pigs were vaccinated.
Okay, mother pigs.
Integrate.
Yes. And if they would like to surveillance ball virus in bats in Thailand, which virus they should be concerned?
Oh, in that project, we unfortunately analyzed the dengue or chikungunya or Zika viruses, but they are distributed in Thailand.
Okay.
So we don't know why cannot isolate these viruses. Maybe the insect viruses are suppressed.
Okay.
Possible, I think.
Okay, so now it's time is out. So thank you very much for Professor Kuwata. So
Thank you very
may I
much.
move to the next speaker.
Thank you very much.
Thank you very much.
For the next speaker will be Dr. Sushawan Pon Sukharom. I will give her a little bit background. Dr. Sushawan Pon Sukharom graduated with from Doctor of Veterinary Medicine in 2008 from Faculty of Veterinary Science, Chulalongkorn University, Bangkok, Thailand. She was granted a full scholarship in 2010 by the Royal Thai Government, Ministry of Science and Technology to study in graduate program abroad. She pursued her master's degree in public health, in veterinary public health specialized at the Ohio State University, United States of America from 2011 to 2013, and she earned her PhD in major in comparative biomedical science at North Carolina State University, College of Veterinary Medicine, North Carolina, in class of 2018. Recently, she works as a faculty member at Faculty of Veterinary Medicine, ราชมงคล University of Technology ตะวันออกชลบุรี Thailand. Her area research focusing on molecular epidemiology of foodborne pathogens, antimicrobial resistance, and bioinformatics. She will talk about, do we need molecular characterization for routine Drosophila diagnosis on clinic? And right now, ladies and gentlemen, I give you Dr. Sushawan Pon Sukharom. Please go ahead.
Can everyone hear me?
Yes.
Yeah, thank you. Oh, thank you for the, sorry. Okay, done. Okay. Thank you for the introduction and good morning. Today, I am really pleased to have a presentation here in the title of Do we need a molecular characterization for routine dilophilaria diagnosis on clinics? The idea of this research came from the 6th-year veterinary student, Faculty of Veterinary Medicine, Rajamangala University of Technology, Tawan Ok, and me, Suchawan Pasuk Arom, the speaker for today. Okay, for the background, I think everyone know about canine heartworm disease. Here, the life cycle of the Dilophilaria immitis, according to the CDC, they are transmitted by the mosquito, and the microfilaria are ingested by the mosquito and become the infective state. At the third stage, larva, right here, the pathogenicity are obstruction, severe lung disease, heart failure, and other organ damage, causing depression, cough, asthma. Also difficult to breathe in dog. Sometimes it can be accidentally zoonosis in human. Not only the Dilophilaria immitis can cause the canine heartworm disease. The Dilophilaria rutens that usually found in Europe area, the Brucia malaya, Brucia hankai, and other genus can be caused the heartworm disease. The mosquitoes such as Mansonia, Anopheles, Culex, Aedes are considered as the biological vector. Not only the dog has been reported at the reservoir, cat is also infected with the Dilophilaria immitis as well. As we know, our Thailand is one of the endemic area of Dilophilaria immitis and Brucia species. Not only that, many kinds of mosquitoes are available in Thailand as well. On the practical clinic, I think most of the clinicians have a routine diagnosis for this disease, such as the microscopic examination that examines the buffy smear and the commercial immunochromatographic test our commercial test kit. This method is had an idea of the ELISA technique. These two methods is usually very easy. And when we got the positive result, we can make sure that the dogs are infected with Dilophilaria immitis. But when we got the negative result, what can we interpret? So it's come up with our question in our research that do this routine detection method adequate for heartworm diagnosis? And the dog can get in the treatment program in time. You know that if the dog get the infection, but we cannot diagnose. So the disease can be go to the severe state and can be the cause of death. So we come up with our working hypothesis that the molecular detection method, such as the polymerase chain reaction or the PCR, can identify canine heartworm disease better than the routine diagnosis on the practical clinic. So we come up with the objectives. The first one, we would like to compare the method of heartworm disease detection on the clinic, including the buffy smear, commercial immunochromatographic test. We use SNAP4DX test for the representative here in our study and the polymerase chain reaction. The extra objective, we would like to determine the suitable kind of the blood sample collect for the heartworm disease detection on the clinic. In here, we use whole blood and the plasma. This picture shows the study design. For the site sampling collection, we collect 113 samples, blood samples from the dog in Chonburi province and Nakhon Nayok province. The reason that why I choose two provinces because you know that Chonburi is a humidity and subhumidity area. It's a lot of rain and maybe we can find the mosquito everywhere and we hope to see the presence of heartworm disease. As well as Nakhon Nayok is very well known of the mountain and river scenery is near Khao Yai. You can see a lot of mosquito and we also hope that we can see the canine heartworm disease there. So we collect the blood sample, both the whole blood and the plasma right here, and we transfer to test for the heartworm disease. The first method that we use is buffy smear. The second one is the SNAP4DX test kit and the PCR. Then we collect all the data and transfer for the utilization. We use the data and analyze and use for all the clinic, and we can plan for the treatment in time, and we can plan for the mosquito control. Here are the material and methods. The first one, the Buffalo smear. We collect the fresh part and we do the Buffalo smear on the glass slide here. We stay with the rising sun and we looking for the microvilli under the microscope. The second method, we use the SNAP4DX Plus test kit. Here the picture shows the result of the SNAP4DX test kit. You can see the spot here on your left-hand side above. Here is a positive control. And when it's positive for the heartworm antigen here, you see the spot on the right-hand side here. This table from the manufacturer index. You see that the heartworm, they show the sensitivity and specificity of the test kit. Here is around 99% of the sensitivity and it's 99.3% for the specificity. They compare with the gold standard test for the positive. They compare with the necropsy method. And for the negative test, they compare with the PET check heartworm PF antigen. You see that it's very high, right? So, for last but not least, the molecular technique that we use in our study is polymerase chain reaction. We're looking for the DNA of dirophilic imides. The primer is specific to the cytochrome C oxidase gene 1, which have the product size around 150 base pair. This picture shows the sequence of our forward and reverse primer according to O et al. 2017. They claim that their primer is 100% sensitivity and specificity to the dirophilic imide species. This picture shows the process of the PCR in our study. Now, we move to the result. What we found in our study. The first one, this slide, is show the prevalence of the positive result based on the location. You can see here on the graph, the x-axis show the province and the overall, and the y-axis show the percentage of the positive prevalence. The light blue bar here, it shows the overall prevalence in our study. It's around 51%. And the back line here, it shows the error bar. You can see here, Nakhon Nayok has around 68% of the positive, and the Chonburi province has around 41 or 43% of the positive. From here, Nakhon Nayok found a significant higher positive percent of dirophilic imide infection more than Chonburi province. Next one, it shows the prevalence of positive result based on the method of detection. Here the graph shows the method of detection on the x-axis, and for the y-axis, it shows the number of samples. The pink one is the positive result, and the blue one is the negative result. Most of the positive result is on the PCR here. It's around 45%, followed by the Buffalo smear is around 15% of the positive here. The last one is SNAP4DX test kit is just 8% right here. And we can transfer to the pie chart here. You can see the area of intersection of each method. Here, the majority of our samples were positive for the PCR. Is around 46 that's positive only for the PCR. And 7 samples is positive only for the Buffalo smear. But the most interesting point is here. These 4 samples, they positive for all 3 methods. You can see that no positive sample is only found on the SNAP4D test kit. No. If you found a positive on the SNAP4D test kit, here, it's also positive for the rest of the method. This slide, it shows the result that according to our extra objective, the whole blood had a significantly higher nucleic acid concentration than the plasma when we measure with the nanodrop machine. But when we work with the PCR or the SNAP4D test kit, they have indifferent in the result. And so both of them work well on our study. However, if you want to work in the remote area, plasma can be preserved longer than the fresh whole blood. Because at the beginning of our study, we faced a lot of the problem that we have to throw away a lot of samples because of blood clot or spoil blood. So we make a decision that if we want to keep for later to work with the blood later or to keep in the refrigerator, we have to work with the plasma. Now I think all of you will get the idea what we are doing in our research and what we get from our research. So we move to the discussion part. The result of the experiment were divided into 3 cases. The case 1, the positive result were from the Buffy smear only. You can see picture on your left-hand side here. It's a microvilli in the bloodstream under the microscope. This one is a true positive. We can make sure that the dog was infected with the Dirophila immitis. But when we move to the right-hand side picture, oh, what is it? We can't make sure that is it microvilli or not, or maybe it's just artifact. It's not easy for us to differentiate the filarial species under the light microscope because this technique has a low sensitivity and requires a lot of filarial infection. If you don't have enough the number of microfilaria, you can see it under the light microscope. Moreover, the microscopic examination is not practical for testing a large number of samples. For the case 2, the results were only on the PCR process. Yes, it can be confirmed that the dog was infected with Dirophila immitis. As we talked previously, our primer is very high sensitivity and specificity to this species. But when we found only the positive for the PCR, but not on the Buffy smear, what does it mean? It can infer that the dog may have an early infection with the parasite but not fully grown, or the number of the microvilli is not much enough that you can see under the light microscope. So when you see the positive on the PCR but not on the SNAP4DX test, the dog may infect with the male Dirophila immitis only. You know that the said kit is very specific to the protein of the female Dirophila immitis. For our PCR, that we have no cross-reaction with the other species. The picture here show the result from the PCR. These are the ladder here. And this lens show the positive control. The bright band here is the product is around 150 base pair. And these two lanes is the negative control. The positive sample is shown here. You can see the band here. And negative result is shown here. You can see any band. For the case 3, the result were positive from all 3 methods. It can make sure that it should be confirmed that this dog was infected for Dirophila immitis, that's for sure. For the Buffy smear, we found the microvilli in the bloodstream. For the SNAP4DX test kit, we found the female Dirophila immitis because, as I told you before, it is a test for specific protein of the reproductive tract of this species. And for the PCR, we found the genetic of Dirophila immitis, which could be confirmed the infection. Here show the picture of SNAP4DX plus test positive. And here, in our study, we found 4 samples that positive for all the methods. The last one, other case. When is the dog are free from disease? What does it mean with free from disease? When? So it could be confirmed that the Dirophila immitis infection if the positive result that on a commercial test kit, but if it's negative, it doesn't mean the free of disease. One interesting point from Little et al. 2018, they proposed that the heat pre-treatment of blood sample can reveal the antigen and can change the result from false negative to the positive for the commercial test kit. But they didn't recommend the heat pre-treatment as a routine on the clinic. For us, we never tried this method before, maybe for later. Same as when we found a negative result from the Buffy smear, it could not confirm the free of infection. And when we found a negative on the PCR detection in this study, yes, it may be clear of Dirophila immitis, but not for the other species of heartworm, because our species, our primer has no cross-reaction to the other kind of the species. According to our research, we can summarize that the PCR is a highly specific and sensitive, but it's costly, time-consuming, and skill required. For the Buffy smear, it's the accessible and the easiest method that take less time and cheap but low sensitivity. For the SNAP4DX test kit and other kind of commercial test kit, is a popular one and take less time, high specificity. We can have a confidence in the positive result, but it is expensive. And some family cannot afford for it and may confuse with the false negative. So right now, I think all of you will have your own decision about the question that I give in the beginning of the presentation. Do we need a molecular characterization for routine dilophiliaria diagnosis on the clinic? For us, our team, yes, we need, because we cannot confirm any negative result from any method. So we need a molecular characterization to help us for the the infection. So you can get the dog to the treatment in time, and you can plan for the protective strategy as soon as possible. And for the next step, for now, we cannot diagnose anymore about the other species of the heartworm disease. So the further study, we would like to study about the prevalence and identification of other Dilophiliaria or Buckia species that found in Thailand. In the future, we can develop the multiplex PCR for heartworm mutation in multiple species. Okay. Acknowledgements. We really appreciate Dr. Warawut Lerk Amnoi Cho, the Dean of Faculty of Veterinary Medicine, Rajamangala University of Technology Tawan Ok, and Dr. Yuvadi Kongpirongchut, Vice Dean for Academic Affairs, that giving us an opportunity to speak here. Many thanks to Veterinary Animal Hospital, Veterinary Diagnostic Center, Immunovirology Lab, and Pathology Lab of Veterinary Medicine, and the Plant Pathology Lab of Faculty of Agricultural Natural Resources, Rajamangala University of Technology Tawan Ok. We love all dogs in our study that devote the blood for us. These are references, and thank you.
Present is about the limit detections of clinical practice diagnosis for dog heartworm, but nowadays she tried to use PCR, which is high sensitivity and specificities on DNA to detect Drosophila. So, any questions from the floor? Okay, one question from the floor. Rely on the test kit SNAP4DX that's used for detection of female adult heartworm antigen. In your opinion, is possible to design the new test kit using antigen of microfilaria instead of clinical case?
Yes, right now I think we can decide more, not only detect for the female antigen, it can be maybe every kind of antigen, but we need time to develop it, and maybe we try to check many kind of test kit, but it can be, maybe it's not same as the IDEX that we use in our study. It's interesting point, and we can make developer only. Maybe I but
Okay, the next question from the chat box. Did you find other filaria worm in Buffy coat smear?
We're not sure. That as I said before, that we need to ask the professional and to confirm what kind of it, because our eyes cannot differentiate the species. Maybe we need the help from the PCR method that can check the other species. Right now, say no.
Okay. Anyone have questions?
My name is Subansa. I have some questions to ask Dr. Suchawan.
Okay.
Hi, Dr. Suchawan. I have a few questions to asking you.
Yeah.
First, due to the SNAP4DX is a diagnosis tool in the clinic, due to my knowledge, is detect the antibody respond to any infection that the tool can detect. So, do you have any suggestion to put the PCR method to the first choice of the diagnosis tool in the practical issue about the cost-effective, time consumer, accuracy, or anything else?
So for first one, from our research, we found that if it's positive on the test kit, it makes sure that it's positive. But what happened with the negative one? Yes, if we see the negative, in our research, we think that not only one method is not enough for us. maybe we should work together with the PCR. I know that the clinician or the clinic, they usually give the medicine before the result of the molecular is come to them. It's good. It's good. It can do in advance. But for right now, I think you can do together with choose one method together with the molecular method to check that free of the disease, truly free, truly free, not a false negative. Should do together. But for the time right now, you know that the PCR is trained to be cheaper and cheaper. Maybe it's good idea that you can work together. You can work together. But the test kit is still what is expensive, everything and expensive. But the PCR is still cheaper right now. It's good idea.
And question number 2, why you choose to study on the COI genes for the skinning primer? Is that a concept region or the anything reason?
For in fact, I'm not looking for the whole process about it. But why we choose the cytochrome C oxidase? Because it's from this paper that O et al. 2017 they said that at high sensitivity and specificity to this species to dirophile images. That's why I choose it. But maybe it's conservative or the other kind of machine. I'm not sure. We have to look for it again. And yes, when you know that when it's very high specific to the dirophile image species, so it can't get any cross reaction to other. But maybe in Thailand, it may be other species also available.
Thank you for your answer.
Thank you.
Thank you very much for Dr. Sushawan. We hope that in the near future we do have like a test kit.
I hope that or maybe to help.
Okay. Thank you again. And we move to the next speaker.
Thank you.
Thank you very much.
For the next speaker is Professor Dr. Chai Wai Lee from Department of Tropical Agriculture and International Cooperation, National Pintung University of Science and Technology, Taiwan. He will be present in a novel approach for detecting and curing subclinical bovine mastitis. And right now, I give you for Professor Dr. Chai Wai Lee. Please go ahead.
Okay. Good afternoon. Can you hear me well?
Yes.
Yes.
Hello?
Yes.
Yes.
Yes, we hear
Yeah.
you.
All right. Okay. Thank you for your invitation for me to give a talk in this web seminar hosted by Kasesa University. And the topic I'm going to talk about is a novel approach for detecting and curing clinical bovine mastitis. And I am working in the Department of Tropical Agriculture and International Cooperation, National Pindong University of Science and Technology in Taiwan. And as you can see that this subject is related to using detection dogs to detect the subclinical bovine mastitis. And before we start, I would like to introduce our working dog training school. This is a unique institute in our university, and actually is probably the only one dog training school in a university in Taiwan. And we train dogs to detect all kinds of different biological targets, including red fire ant, termite, and or other bacteria. I was the director of this training center, and that's why I was trying to have an idea that how can I apply these dogs for detecting mastitis, because which is my expertise. Okay, so let's start with a few slides talking about subclinical bovine mastitis. Since it's subclinical, so it's difficult to detect unless you perform somatic cell count or you do biotech bacteriology to identify the presence of bacteria in the milk. And the pattern of inflammatory infection of mastitis is like this. So in the healthy cows, you have non-infected quarters. So if the cows are exposed to pathogens, and also if the cows are like under the condition that they are susceptible to the infectious pathogens, they would develop a subclinical infection. And at this stage, it's kind of like for the cows to have the immune system and the invading pathogens that are combating against each other. So if the immune system wins, the quarter or the infected mammary gland is going to have a spontaneous recovery and back to healthy status. In contrast, if the immunity is not able to control the invading pathogens, and it will develop to clinical quarters. So at this stage, we are going to be able to see the symptoms. And however, at the subclinical stage, we cannot see any symptoms unless we analyze the somatic cell count or the presence of bacteria. So in most of the commercial dairy farms, when they see symptoms, they would treat the cows with antibiotic therapy. And after the therapy, the infected quarter might come back to a subclinical status, or they might completely recover to a non-infected quarter. So the problem of subclinical mastitis is that it's invisible. So therefore, we don't a lot of milkers don't even know they exist. So the reduced milk production is which accounts for 70% of the economic losses of mastitis if we don't treat them. And in this slide, it's just it's just for giving you an idea that when we only look at clinical cases, they are visible, right? So we can see this as an iceberg. So the clinical cases are above the sea level. But however, every case of clinical case underneath the sea level, there are a lot of subclinical cases. So it has been estimated that a clinical case, behind one clinical case, there are at least 20 subclinical cases which are invisible. So if the dairyman only look at the clinical case, then he might look happy because there might be only 1 or 2 cows show clinical symptoms. However, if they could see the both subclinical and clinical mastitis cases, this is how they would look like. You're going to see a lot of cows, they are actually got the infection with mastitis. It's just that they are subclinical. So we have research analyzing the cases of mastitis, and we actually found that clinical mastitis is a lot. I mean, the percentage of clinical mastitis is a lot lower than that of the subclinical mastitis, which is exactly the same as we described before. And the incidence of subclinical mastitis was investigated in 9 dairy farms if I remember correctly, it was in Finland. And then we could find that at the cow base, the 36.7% of the cows are infected with subclinical mastitis, and at the quarter base is 15.7% of the quarters are infected with subclinical mastitis. And most important, they are invisible. And almost all kinds of bacteria can induce mastitis, and then depending on the management system, depending on the geographical location. And then in this study, we could see that most of the subclinical mastitis cases are caused by Staphylococcus aureus or coagulation native Staphylococcus, right? And many of the Streptococcus species. And different locations would, due to different management system and different climates, they would have different profile of these bacteria that are able to cause subclinical mastitis. Okay, so what are the impacts of subclinical mastitis? First of all, it's gonna reduce the price of milk, because in most countries, the price of milk is decided by the level of somatic cell count. And when cows are infected with subclinical mastitis, the milk production is going to be reduced. And on top of that, because they are invisible, so they are not isolated and they are not treated, so they will become the reservoir of infection in the herd. And once they turn chronic mastitis, the culling rate of these animals is increased. So it has been estimated that the 25 to 30% of subclinical mastitis will turn chronic, and they are very low response to antibiotic treatment. And I have talked to dairy farmers in Taiwan, and I was told that when they use antibiotics to treat mastitis, the curing rate is probably only 10%, which means that due to the abused administration of antibiotics in the past decades, the bacteria that inducing mastitis have developed a great resistance to the antibiotics. And it's not only the milk production for the current lactation. A cow that is infected with subclinical mastitis would have the milk production of next lactation affected, and it could be like decreased by 9.1 kilo per day, according to a study conducted in 2008. And so over here, we can see that as the biotank somatic cell count increases, the average milk yield loss is increased in proportion. Okay, so after analyzing 164,000 of cows in 22 herds, like 60 days after the pasteurization, we acquired these information. So each of subclinical mastitis that had a reduced milk production, about 720 kilo, which is equivalent to $285 per case. And the chance of having subclinical mastitis increased 2.5 times. And the chance of being cowed due to the turning chronic mastitis increased 3 times. And having the subclinical mastitis also delayed the successful conception. And in general, or averagely, it's delayed by 17 days. Okay, so traditionally, we have a couple methods for diagnosing subclinical mastitis. The basic way is to do bacteriology. So we have to plate the milk and to find out what are the bacteria present in the milk. And about 10 years ago, we have published a paper. So we developed a microchip, a biochip that can detect the presence of these bacteria in a few hours. And by plating the bacteria, it will take at least 18 hours to know the results. And on the farm side, we can do this so-called the California mastitis test, CMT. And this is a very simple way, but it's not that accurate. All right, so by mixing the reagent with the equal amount of milk, and then we look at the characteristics of the mixture. So it can be divided into negative, trace, weak positive, distinct positive, and strong positive. So depending on the degree of gel forming that in the mixture, if it's become like very sticky, that means that the somatic cell count is very high. And recently we have this a more advanced machine that is able to analyze the somatic cell count, and this is actually what I'm using. But the cassette is quite costly. Each of the cassettes costs $3 to $4. So that means that if you have a lot of samples to analyze, it's gonna be quite costly. But the accuracy is acceptable. Yeah, I have compared the number of somatic cell counts like analyzed by this machine with the large machine, and the difference is within 5%. Okay, and if the milking machine is more advanced, it actually has the built-in facility to measure the electrical conductivity, because mastitis increases the amount of sodium and chloride, which is going to increase the electrical conductivity. So as we can see in the figure, in healthy cows, the electrical conductivity is pretty stable. However, when cows got mastitis, the electrical conductivity in the milk is gonna fluctuate a lot. However, this is not 100% accurate. All right, so because normal milk is like 5.4 to 5.6 millisiemens a centimeter, and for subclinical mastitis, depending on the cause of bacterial strains. For Staphylococcus aureus, the electrical conductivity is increased dramatically, but for Streptococcus urbaris, it remains almost the same as normal milk. So it's not 100% accurate. So in summary, we can know that for detecting subclinical mastitis, it's very tedious, and it's almost impossible for the commercial dairy farm to do the CMT or to do bacteriology like every week or on a regular basis. So that's why I had this idea to use the detection dogs. So we train the dogs to detect the bacteria that can cause subclinical mastitis, and then see if we can just use the dogs to detect the mastitis. So first of all, we have to have this cognition of sense training. So we use 3 beagle dogs to detect 3 targets, including E. coli, Streptococcus aglexia, and Staphylococcus aureus. So when dogs did or identify the targets, we train them to sit down still as a passive response. And then we also encourage them to search the targets independently. And training dogs to distinguish targets and non-targets is also important. So in addition to the 3 strains of bacteria that are causing mastitis, we also train them not to respond to bacteria not inducing mastitis. And this is important for them to distinguish targets and non-targets. So in this study, positive reinforcement training method was used, which means that when they have a correct response, we give them treats or food phrases as reward, so that they could perform or distinguish the targets more correctly. And before we go further, I need to explain a few terms that we're gonna be used later on. For sensitivity, it's like the positive response to our target. And specificity is like no response to the non-target. All right, so they are different. Sensitivity is like the correct detection of and give us a positive response on the targets, meaning the milk containing mastitis causing bacteria. And specificity, on the other hand, is that we give them like milk without bacteria, and they are not supposed to have response. And the accuracy rate is the number of corrected responses, including like targets and non-target, divided by the total number of samples. Okay, so these pictures shows like how we train the dogs. So we have these the samples put in the cans, and then we need to have a trained person guiding the dogs to sniff the sample. And when the dogs detect the target, they sit down still like this as a positive response. So we have many samples like arranged in this room, and the dogs are sniffing the samples one by one and give us a response. And they can do like 30 samples, for example, like in just like within 1 minute, because their sniffing ability is very sharp. So we have a person like guiding them to sniff the samples one by one. So this is very fast. And after we train the dogs, we need to see if like they have a high sensitivity to identify the bacteria that we have trained them. So we have 3 dogs, and their IDs are A, B, C respectively. And then we can see that the accuracy rate is very high. It's almost 100%. Okay, it's only for dog C. Like it had a false response on the Streptococcus once. So basically, we can know that these dogs have very good sensitivity to the targets. And then we started to train them more. So we do the experiment, which is a double-blinded experiment. So both the guider and the dogs do not know that the location of the targets. And when we finish a round, the targets are rearranged, and then we do these 5 rounds a day for 8 continuous days. So in each round, we have 3 targets which contain 10 to the power of 7 CFU of the bacteria and the 17 non-target tubes. So therefore, each dog would contact 15 targets per bacteria strain and 85 non-target per day. So this was done by 8 days. And as you can see that for E. coli, for strep, and for staph, the accuracy rates are all above 98%. And then we started to investigate like what would be the detection limit for these dogs. So our bacteria sample are serial diluted, like 10-fold diluted. So we have then diluted like 10 times, 100 times, 1,000 times until like 10 to the power of 10. And surprisingly, even though they are diluted like 10 to the power of 8 or 9 or 10 times, they can still have a pretty good accuracy rate. Of course, like when these samples are diluted a lot, the accuracy rate is reduced to 88%. But for all the 3 strains of bacteria, and when we have an undiluted sample to samples that are diluted by 10 to the power of 10 times, the accuracy rate is still like around 95%, which is still acceptable for our purpose. So at the the last stage, we wanted to test if these dogs are able to detect milk with subclinical mastitis from the field. So totally 51 samples were collected from a commercial dairy farm like around the campus, and then we had these samples analyzed to confirm that they have a high somatic cell count, and also there's a presence of mastitis causing bacteria in the milk. And we also found the samples with cephalococcus and streptococcus because normally E. coli does not induce subclinical mastitis. E. coli induce acute mastitis. And we have 3 dogs, and averagely the sensitivity and the specificity of the 3 dogs was 90.2% and 90.5% respectively. So that means that we can actually apply this like to the dairy farm, like commercial dairy farm. So we want to work together or cooperate with the dairy farms. So then I'm trying to talk to some dairy farms so that they can have their milk samples like collected in tubes and have them delivered to our working dog training school. And we would use the dogs to sniff the or to detect the cows that with subclinical mastitis, and then we were gonna go back to tell the farm that okay, how many cows and what are the cow numbers that might have subclinical mastitis, because the accuracy rate is is above 90%, and it's quite interesting because the reason that we only have a 92.2% of sensitivity, it's actually higher because when we tested these samples from the field, there were a couple of samples. The dogs got the positive response, that showed positive response, but we checked the somatic cell count and the presence of the bacteria. Both of them were negative, but the dogs had a positive response. So then we thought this was just a false alarm. But after a few days, these cows developed the subclinical mastitis. So it could be concluded that the dogs are actually able to detect the subclinical mastitis earlier than the increase of the somatic cell count, and also even the presence of bacteria. Also, the sensitivity is very, very high. All right, so this is a part for detection. And then the second part is for like once we find these cows with subclinical mastitis, how are we going to treat them or cure them? And I have been developed a product with a company with that. It's called the Other Fresh, and this is a nature product. So we want to use the dogs to have an early detection on the subclinical mastitis, and we want to use nature treatment to cure the subclinical mastitis. So this product is highly safe because it contains neutral nature extracts, and mostly from creatures that are from like high mountains, plains from the high mountains, or creatures from deep sea water. So to the immune system, the degree of foreignness of these particles is very high. And then we also use sodium arginate as the carrier. And so this product enhances the innate immunity, which is non-specific. So it no matter the cows is infected by Staphylococcus or Streptococcus, the innate immunity is going to clean them up by the host. And this product was developed based on the theory of homeopathy. which means that only extremely low dosage is required. And homeopathy is based on a traditional medicine therapy that was developed in the 1770s from in Germany. And the theory is based on like cures like, which means that if a substance causes a symptom in a healthy person, given that person a very small amount of the same substance may cure the illness with that symptom. So in theory, a homeopathic dose enhances the body's normal healing ability. So when we use this product to cure subclinical mastitis, there's no concerns of antibiotic residue. All right, so the product looks like a syringe, and it contains like 30 millimeter of the reagent, and then with this cannula that can be infused into the mammary gland that with subclinical mastitis. So before we start to use this arteflush to cure subclinical mastitis, we wanted to know if cows have response to this arteflush, because according to the theory of homeopathy, we infuse this arteflush to healthy quarters, which only has 160,000 somatic cells like per milliliter, and within 1 day it was increased to 2.8 million, and gradually it decreases, and on the day 7 after the infusion it came back to normal. So it only induced mild inflammatory responses, and it increased leukocyte migration from blood to the milk. So that's why the somatic cell count is increased dramatically, and the bactericidal capacity of these leukocytes is also increased, because when we have inflammatory responses, a lot of cytokines that are secreted, so that the killing capacity of these leukocytes is increased, and this would eventually facilitate the clearance of the pathogens. So when we infuse the arteflush into healthy quarters, this is what we see. So the somatic cell count will come back to normal within a week. And the next stage is that we at the selected 10 cows that are diagnosed with subclinical mastitis. So they had somatic cell counts anywhere between 700,000 to almost 4 million, right? And then we infuse the arteflush into the quarter, and after day 1, day 2, until day 7. So at day 7, 6 out of the 10 cows, 60% of the animals that treated cows had the somatic cell count reduced to less than 400,000. So this is within the range that it can be sold to the milk processor. In Taiwan, the limit is 500,000. So the average somatic cell count of treated cows was reduced from 2.3 million to 625,000 somatic cell per milliliter. So in this, from these results, we know that using this arteflush, which is a natural product, is able to have a pretty good curing rate on the subclinical mastitis cases without using antibiotics. And we would like to promote this product, and so that the farmers, actually the farmers in Taiwan, they are very careful about the antibiotic residue in milk. So therefore, more and more farmers are using this product. So in conclusion, the diagnosis of subclinical bovine mastitis is very time consuming. It cannot be practiced by the farm on a regular basis. So therefore, the presence of subclinical mastitis are normally being ignored and not treated. And scent detecting dogs can be trained to detect mastitis causing pathogens in milk with pretty high sensitivity and specificity. So the results of treating mastitis at the early stage, hopefully in the future, we can use the dogs to do the job for us. With the nature and the immunostimulating formula such as Artefresh, it's satisfactory. It's the curing rate is about 60%, and this is actually even much higher than using antibiotics. So combining an earlier and easier detection of subclinical mastitis with Artefresh may be a feasible approach to minimize the antibiotic administration and the resistance, so that the quality and the safety of dairy products can be increased for the consumers. Thank you very much.
Including chloroform, staphylococcus, and also streptococcus. So this is the interesting topic. So now the time is run out, but okay, one question from the floor. Okay, one from the floor is that is the dog detection, this detection is vary by dog breed, dog age, or dog behavior?
Yes, it's not like, of course, it's not that all the dogs are able to be used as detection dogs. And according to our experience, it's probably only 1 out of 10 dogs is suitable to be used as scent detecting dogs, because one thing is that the dog has to be very curious. Okay, so the curiosity of the dog has to be very high. So he has the motivation to sniff anything around. And the second thing is that the dogs has to be very greedy on treats and foods. That's why when we give them rewards, they are encouraged to do the job. Okay, and the third thing is that the dog has to be successfully socialized, which means that the behavior of the dog should not be interfered by sound or things or people's around. Okay, so it's not easy to find a suitable dog for training.
Okay, so thank you very much for Professor Lee.
You're welcome.
Thank you. And right now we're gonna take a picture together on of the invited speaker and on of the participant. Could you turn on your camera on your computer that we can take a picture together, please? Are you ready? Just take a few minutes. Okay, thank you so much
ครับ
for
ครับ
your
ยังไง
time. And for the
ครบ
next
ใช่ไหมครับ
speaker,
Okay.
that is Professor Dr. Dionysios Mentavinos from University of Patras, Greece. Before he give us a talk, let me give you information for his background. Professor Dr. Dionysios Mentavinos served as a professor of wastewater engineering in the Department of Chemical Engineering, University of Patras, Greece. He holds a diploma in 1991 in chemical engineering from Aristotle University of Thessaloniki, Greece. And he earned master degree and PhD degree in chemical engineering from Imperial College of Science, Technology and Medicine from United Kingdom. After a short spell as a research fellow and lecturer at Edinburgh and Leeds University, respectively, he returned home to take up an academic position as an assistant, associate, and full professor in the Department of Environmental Engineering at the Technical University of Crete before he moved to University of Patras. His research interests focus on wastewater treatment by advanced oxidation technology, as well as on environmental catalysis. Professor Mentavinos served as editor-in-chief to the Journal of Chemical Technology and Biotechnology and associate editor to the Chemical Engineering Journal Advanced. His publication record include 250 journal papers, over 18,000 citations, and his H-index of 73. Professor Mentavinos is the Vice Rector of Academic and International Affairs of the University of Patras, while he has served as Vice President of the Hellenic Open University between 2019 and 2020. And today he is going to give a talk about the degradation of emerging microcontaminants by electrochemical and sulfate radical based AOPS. And right now, ladies and gentlemen, I give you Professor Dionysios. The floor is yours.
Good morning, everyone. I hope you can listen to me. Is everything fine?
Yes, I can hear your voice. It's very
Excellent.
clearly.
Thank you. Thank you very much. Well, good morning from Athens. It's 7:25 in the morning here. I would like to thank the organizing committee for inviting me to give this talk in your conference. Perhaps you are going to listen something slightly different from what we have heard so far. So the talk of today has to do with the use of advanced oxidation processes for the degradation of emerging microcontaminants of pharmaceutical origin. Basically, we are going to focus on the so-called advanced oxidation processes. They are a family of common, similar but not identical processes, mainly physicochemical, including heterogeneous photocatalysis, ozone oxidation, electrochemical oxidation, Fenton and the like reactions, thermochemical processes, as well as various irradiation sources like gamma rays, ultrasound, or visible light. These processes share a common feature. This is basically that make advantage of the production of powerful oxygenated oxidation species, what we usually call reactive oxygen species, that participate in the oxidation mechanism. Such species are mainly, but not exclusively, free radicals, and the dominant, but not the only one, is the hydroxyl radical. We've also got sulfate radicals, because we are going to talk about them today, as well as superoxide radical anions, and many more species that are not radical, such as the photogenerated conduction band holes. just to get an idea of how strong these oxidants are, this list shows the oxidation potential of various species with hydroxyl radical being second only to fluorine, closely followed by sulfate radicals. This list also includes ozone, hydrogen peroxide, permanganate, and so on. So, to answer the question, why have advanced oxidation processes been researched, developed, and partly applied to in the past 20 or 30 years? The answer is fourfold. Firstly, for the degradation of organic microcontaminants in waters and wastewaters, like pesticides, pharmaceuticals, endocrine disruptors, natural organic matter, disinfection by products. And this actually is the focus of this talk today. Second, for the treatment of industrial effluents that contain non-biodegradable species, like agro-industrial effluents, textile industries, paper mills, hospital effluents, landfill leachates, refineries, and so on. Third, although this is not very common, yet for the deactivation of microorganisms, in order to replace classical disinfection methods like chlorine in waters and wastewaters. And last, four, it's a rather case-specific application for the oxidation or reduction of metals or metalloids to less toxic ions. Standard examples are the reduction of hexavalent chromium to the less toxic trivalent one, or the oxidation of hazardous arsenic-3 to less hazardous arsenic-5. Today, I'm going to talk about two such processes. The first one is electrochemical oxidation. It's a quite old advanced oxidation process. Actually, it dates back to the 19th century when the electrochemical decomposition of cyanide was investigated. It is a process that has been used for various applications besides environmental remediation. However, for purposes of environmental remediation, extensive investigation goes back for about 40 or 50 years or so. The setup is quite simple, straightforward. What you need basically is an electrolytic cell which consists of two electrodes. The anode, where oxidation reactions take place. Basically, this is where oxygen evolution from water splitting occurs. And the cathode, where reduction reactions take place. This is where hydrogen evolution occurs. Currently, the system is supplied by electricity. And of course, you've got here your liquid phase, let me call it electrolyte or wastewater. The trick is that your liquid phase to be treated has to have considerable conductivity in order to get the current going around. So, the major features of the process are as follows. The great advantage is that we are talking about a clean technology in the sense that no extra chemicals, except perhaps the electrolyte, which is some kind of salt, basically, are needed. And there are various materials that can be employed as anodes or cathodes. Apparently here we are mainly interested in the anodic material because this is where oxidation takes place. So we've got a variety of the so-called active dimensionally stable anodes like titania, iridium, platinum, and so on. And more recently, we've got a new class of anodes, the so-called boron-doped diamonds, BDD, which is a non-active anode. The main drawback is that usually it costs a lot because you pay electricity to power the system. Regarding the mechanistic aspect of the process, electrochemical oxidation occurs through two major pathways, a direct one, which I'm going to talk about in the next overhead, and an indirect mechanism. The indirect mechanism requires the formation of secondary oxidants, and such oxidants may be chlorine or chlorine-containing organics, sulfate radicals, ozone, or hydrogen peroxide. However, the heart of the process is the direct oxidation mechanism. So what basically happens on the anode, this is where physically adsorbed hydroxyl radicals or chemisorbed active oxygen is formed. Basically water and M is the metal of the anode. Okay, there is some kind of a physicochemical process and you get this sorbed hydroxyl radical, and this is the first stage of the process. If I wanted to specify this mechanism in the non-active boron-doped diamond, this is what we are going to use in our experiments today. Those interactions between the hydroxyl radical and the electrode surface are rather weak, which means that the formed radicals can easily be liberated and are available to react with the organic matter R, okay? And this apparently results in some kind of deep oxidation, mineralization, or total oxidation. So basically, you go quite fast to carbon dioxide and water. So non-active anodes, unlike active anodes, promote total oxidation reactions, while active anodes promote mainly partial oxidation reactions. The drawback, again, is that there always is a side reaction that competes with the actual environmental reaction of hydroxyl radicals, and this is what we usually call as oxygen evolution reaction. So instead that the absorbed radical reacts with the organic matter, liberates oxygen, okay, which is an unwanted, undesirable reaction in the sense that we pay money, we consume electricity for something that we don't care whether it happens or not. The second system we are going to discuss today is the so-called sulfate radical SR advanced oxidation process, which basically comes from the so-called Fendon reaction. It's a Fendon-like system, and perhaps the Fendon reaction is the oldest advanced oxidation process. It's a mixture of hydrogen peroxide and ferrous ion at acidic conditions, and this reaction occurs, creating hydroxyl radicals. By analogy, if hydrogen peroxide is replaced by a persulfate salt, okay, iron and other means, that's why we call it activator, can convert the persulfate salt to sulfate radicals, which are slightly less oxidative than the hydroxyl radical, but they are more selective. Hydroxyl radical is a very unselective oxidant, and they have a longer lifetime than hydroxyl radicals. So this Fendon-like process basically can be activated by several different methods. One of them is the use of transition metals like iron and many more. It can also activate it by high temperatures, by heat, as well as by various irradiation sources like ultraviolet light or ultrasounds or gamma irradiation, and so on. So the overall reaction is something like this. Activation of persulfate salt gives sulfate radicals, and as a next step, sulfate radicals themselves react with water, producing again hydroxyl radicals. So basically, it's a mix of two kinds of radicals. You can never avoid in this life the presence of hydroxyl radicals in your system. Now, today I'm going to talk about the degradation of dexamethasone in the liquid phase. Dex is an anti-inflammatory drug. Basically, is a cortisol derivative, and it is detected in hospital effluents, city sewage, river waters at the low microgram per liter level. Electrochemical oxidation will take place on a boron-doped diamond anode with either stainless steel or carbon cloth as the cathode. I don't care too much about the cathode. Anode is the critical electrode here. The concentration of DEX is followed by high-performance liquid chromatography, while the gradation byproducts are analyzed by liquid chromatography, high-resolution mass spectrometry. This graph here shows the effect of applied current density from 0.02 ampere per square meter to 0.2 ampere per square meter on DEX degradation. This is the normalized concentration over the electrochemical oxidation time. The greater the applied current density, the greater the reaction rate, which is reasonable, which is expected, because at higher current densities, more hydroxyl radicals are produced, and this apparently facilitates the contaminant degradation. The next graph shows the effect of initial contaminant concentration on its own degradation from as high as 2 milligram per liter concentration to as low as 2.25 milligram per liter. Again, we can see that the higher the initial concentration, the lower the degradation rate. Apparently, at 2 milligram per liter, it's only 75 degradation after 45 minutes of reaction, while for the other three initial concentrations, we've got complete removal after 45 minutes of reaction. However, what is more important in this kind of studies is the effect of water matrix on degradation. The experiments so far were conducted in UPW, ultra-pure water, but this is an ideal scenario. In real-life applications, you've got this effluence either in WW, wastewater, or in drinking water, bottled water, BW, or so on. So this graph here shows what happens when the reaction occurs, not only in ultra-pure water, I'm talking about the black squares, but in bottled water, open squares, the purple ones are wastewater, as well as in ultra-pure water that has been spiked externally with various species that are inherently present in waters, like bicarbonate, which is the dominant anion in drinking waters, or chloride, or humic acid, HA. Humic acid is a representative of the natural organic matter typically found in waters and weightless waters. You see here, and more clearly on the right-hand side graph, which shows the apparent rate constant of these reactions, assuming that degradation follows a first-order reaction, you see that basically none of the matrices, apart one, affect the degradation process. This is the rate constant for ultra-pure water, far left, and all other bars, except those three in the circle, okay, show similar K values for all other matrices, which is good, because usually the water matrix has a detrimental effect on the process because you've got side reactions and competitive reactions by other species present in water. The interesting thing, but it's something that we rather expect to happen, is what happens in the presence of chloride in water. These are three different concentrations of chloride. You get much higher degradation rates, and this is because electrogenerated chlorine species, okay, like hypochlorous acid or chlorine dioxide or chlorine radicals and so on, contribute to the so-called indirect secondary oxidation mechanism. And then the next step is to use sodium persulfate for the sulfate radical process. Anyway, we want to have some kind of electrolyte in our system to make things happen faster. Okay, so why not use sodium persulfate, which is an electrolyte? So let me go through this graph here. The purple curve here shows how sodium persulfate degrades contaminant in the absence of current. SPS itself, like hydrogen peroxide, is a mild oxidant. Then the graph here, the closed, the black square, is what happens only with current electrochemically without SPS. And the other three curves is when I combine the two individual processes. I get an enhanced degradation rate. And actually, I can quantify the effect of process synergy through this formula up here. Synergy is basically defined as the rate constant of the combined process minus the sum of the rate constants of the individual process divided by the combined process. And basically, if this value is equal to 0, then this shows that the combination is simply cumulative, is simply additive. 1 plus 1 is equal to 2. However, when this value is positive, is greater than 0, then we are talking about real synergy. That is, 1 plus 1 can be 3 or 4 or whatever. Okay. In some cases, although not very frequent, we may have a negative value. This means a competitive antagonistic effect between the individual processes. In this case, this is about 60%, so I've got some good synergy between electrochemical oxidation and persulfate activation. Another thing is that an interesting finding, all experiments so far were on a stainless steel cathode. At some point, we replaced this with carbon cloth, which is a new material for cathodic applications. And what happens is that it works much better than the stainless steel cathode. And the reason for this is that carbon cloth alone is an adsorbent. So this is the experiment up here, where you've got carbon cloth without electrochemical oxidation, without applied current, and you get something like 35% contaminant removal simply due to adsorption. And this also is demonstrated in the right-hand graph, showing the rate constants for the pair carbon cloth and stainless steel. The first two bars are in ultra-pure water. The other two here are in bottled water, and the last two are in wastewater. Carbon cloth performs better than stainless steel, perhaps with the exception of wastewater. And again, this is reasonable to happen simply because wastewater is the most dirty matrix, because it contains various organic and inorganic species. And this debris perhaps blocks the active sites of carbon cloth. And this is why there is no serious difference between the two cathodes in the actual wastewater. These are the 7 transformation products that have been detected during the degradation of dexamethasone. And these are common for all 3 processes studied, that is boron-doped oxidation over stainless steel, boron-doped and carbon cloth as the cathode. And this is oxidation on stainless steel and boron-doped diamond with SPS. The 7 products, intermediates, are common. What is different, as you can see, is their temporal distribution, and this is reasonable to happen simply because the processes, the 3 processes, occur at different rates, at different reaction times. This is an idea of the structure of the 7 identified products, and this can give us an idea of the kind of pathways and reactions that take place. They can be classified as hydroxylation reactions, oxidation reactions, decarboxylation and recompany, and eventually, in the end, fluorine release in the liquid phase. I've got a minute or so, perhaps, or I'm done. Okay, that's my last slide. I would like just to spend a minute or two to discuss this chlorine thing and how important it can be in your system, they will show you another example. This is ethyl paraben, it's an endocrine disruptor, and this has been degraded by boron-doped diamond with sodium sulfate as an electrolyte. But nothing really fancy happens here. You've got hydroxylation and dealkylation reactions, mainly through hydroxyl radicals. Okay, so now I'm going to show you what happens in this system when the electrolyte sodium sulfate is replaced by sodium chloride. Okay, besides the fact that things happen much faster, I've got a totally different distribution of reaction by products. If you see here the upper part of this graph, here you've got a number of chlorinated, you see here and here and there byproducts, and these chlorinated byproducts are by default more ecotoxic than non-chlorinated byproducts. And again, more interestingly, you get a number of dimerization and trimerization reactions that you don't get if you use another electrolyte. So sodium chloride in your electrolytic system can be favorable in terms of kinetics of reaction rates, make things happen fast, but please bear in mind that the presence of chlorine results in the formation of organochlorinated secondary byproducts that might be more toxic than the parent compound. Thanks again for your invitation, and hopefully I would like to be able to welcome you to the University of Patras with physical presence at some point in the near future. Thanks for your attention.
Can you type your question on the chat box and I can deliver to Professor Dionysios, please? First question from Associate Professor Dr. Chanat.
Can you hear me?
No.
Yes.
Yes. Now I can hear you.
I have a quick question about a boron-doped diamond anode. Do you is it commercially available, or do you synthesize it in your laboratory?
It's commercially available.
Is it commercially available? Is it very expensive? I mean, could you just roughly tell me the price on that material?
A pilot scale setup, okay, with almost everything, it's no more than a couple of thousand of dollars.
I see. Okay, next question.
It's not as expensive as it sounds due to the term diamond. Okay, it's not that bad.
Okay. Next question about the did I say it correctly? Dexamethasone, right?
Yes.
Is it easily soluble in water or not? It looks like it's steroid or something. It might not be easily soluble.
At the concentrations of environmental concern, which is at the microgram per liter level, yes. For experimental reasons and for analytical reasons, we are going slightly higher. We are going to the low milligram per liter level in order to be able to do the analysis properly. It's still okay. Yes, at higher concentrations, it's not easily dissolved in water, but we don't want to work with higher concentrations because they are not environmentally relevant. We want to be as close as to the actual problem. Okay. Even working at the low milligram per liter level, 0.5 or 1, we are still higher than the actual level of this contaminant in real waters.
Okay, great. Thank you so much.
Hello. Okay. One question from me, please. According to your experiment from removing dexamethasone by using the coupling technique between electro-oxidation and sulfate and persulfate, you get a good result. And I'm not quite sure what kind of the majority of free radical between hydroxyl or sulfate radical to destroy the structure of dexamethasone. I'm not quite sure. Did you do the experiment for the trapping experiment to prove what kind of the free radical?
Yes, we haven't done any trapping experiments using EPR, electroparamagnetic resonance, because we don't have free access to this equipment, and it's a laborious experiment. However, we have done some kind of indirect scavenging experiments using methanol and butanol, because these usually trap hydroxyl radicals or other radicals like sulfate at different rates. So from this kind of trapping indirect experiments, you can get an idea, but not absolutely quantifiable idea of the two mechanisms. And it seems that both radicals participate in the reaction mechanism, both sulfate and the hydroxyl radicals participate. However, to be able to quantify how much is hydroxyl radical and how much is sulfate radical, you need EPR, that's for sure, electroparamagnetic resonance analysis.
Thank you so much. Is there any question again from the floor? Okay, and thank you so much for your talking from Professor Dionysios from University of Pratas, Greece. Thank you so much.
My pleasure. Thanks a lot for inviting me.
Okay, and right now we have been listening to 7 distinguished speakers. Now is the perfect time for the break for the lunch time, and we will be back again at 1:45. And for the first speaker on the afternoon session will be Professor Dr. Andrea Koshwan from Faculty of Chemistry and Chemical Technology, University of Ljubljana, Slovenia. She will give a talk in the topic of advanced oxidation process for removal of pharmaceutical from waters and wastewater, trend and perspective. Please come back again at 1:45 in the afternoon. Thank you. Hi, good morning.
Hello, good morning. How are you?
เอ๊ะพี่หนึ่งเหมือนเสียงเบาอ๋อ
Can you hear me?
Yes, I can hear you.
Okay, I can also hear you. So may I try to share my presentation?
Yes, that would be great. You can share it right now. We have 5 minutes left.
Yeah.
You can test it
Share.
right now.
So you are seeing my presentation?
Yes, I do see your presentation. It's
So,
all
and it
clear.
is moving back and forward.
Yes.
Okay, so everything seems to be perfect. So I will just wait. Okay.
Okay. We only have 5 minutes left and we will start the meeting again. Thank you so much for your help.
Okay, thank you. That's in the bomb, in the bomb. Ja. Ne, Cisco, ja. Давай на то на друг монитор дать не се квадраш не бom okay се Ja. Ja, zaradi tega, ker je to veterinarska fakulteta. Mene so pa povabili zato, ker delam na
พี่หนึ่งให้ผมพูดได้เลยไหม Welcome back to the afternoon session. For the first speaker in the afternoon section is Professor Andrea Goswan from University of Ljubljana. She will give us in the topic of advanced oxidation process for removal of pharmaceutical from waters and wastewater trend and perspective. Before she giving her topic, let me give her background for one of the audience first. Professor Andrea Goswan obtained her PhD in the field of chemical engineering. She is a professor of environmental engineering and she also is the Dean of Faculty of Chemistry and Chemical Technology of University of Ljubljana, Slovenia. She is also the head of the Program Council of Doctoral Study on Environmental Protection at University of Ljubljana. Her research work is focusing on management of solid waste, biodegradability, and ecotoxicity studies with chemicals and wastewaters in aquatic compartments, risk assessment, and hazard identification of chemicals, as well as different aspects of wastewater treatment, recycling, and reuse. She participated in 6 national research projects. She was a leader of 5 bilateral international projects, and she is currently involved in national research program chemical engineering. And this is the perfect time. Ladies and gentlemen, I give you Professor Andrea. The floor is yours.
Thank you very much. So you can hear me. Everything seems to be fine.
Yes.
Okay. Thank you very much for this very kind introduction. And I would like to emphasize that I'm very honored to be invited to speak at your conference and deliver this lecture. As it has been told, I come from University of Ljubljana, which is the biggest university here in Slovenia, from Faculty of Chemistry and Chemical Technology. And my background is related to chemical engineering. And today I would like to give you a brief overview of advanced oxidation processes for removal of pharmaceuticals from waters and wastewaters. And I will present some main trends and perspectives for the future. I don't know why it doesn't want to move. Okay, sorry. So here is the brief outline of my presentation. I will briefly talk about antibiotics, which are persistent and biologically active micropollutants. Because the conventional treatment methods are not able to remove them from the waters and wastewaters, I will explain a little bit about advanced oxidation processes as viable options for their removal. I will focus on how to monitor efficiency of the processes, and I will use this opportunity to briefly present you our research work in the field of different antibiotics, with case study of thiamulin, which is persistent antibiotic used in veterinary medicine, so it's probably more familiar to you than to me. First, if we have a brief overview of the presence of the pharmaceuticals in the environment, here in the top table, we can see that in the wastewater, in the influent and also in the effluent of the municipal wastewater treatment plant, we can find a lot of different micropollutants, as well as resulting from pharmaceuticals used in human medicine, and also, of course, different compounds which can be found in the plastics and all other items we are using daily. And we can see that conventional treatment plant is able to remove only a limited extent of such pollutants, because here in the last column of the upper table, we can see that the removal rates in conventional wastewater treatment for these compounds can be as low as 30% or 22%, and it can go up to almost 100%. In the lower diagram, we can see that the compounds which were found in the surface waters of the European Union are also very different. So when they started to search different compounds, some of them were found almost in all of the samples, because here the frequency of appearance means that in 100%, that almost all in the samples, these compounds were found. And also in this table, we can see the occurrence of these compounds in different European rivers, and we can see that they are very often found almost everywhere. So that means that we have a problem regarding these pharmaceuticals. Antibiotics is a specific group of pharmaceuticals which are quite often used in very large quantities, are not only beneficial because they can come into the environment and affect a variety of non-target organisms in our environment, from microorganisms to higher organisms as well as plants. If they're present in the environment in very high concentration, they can, of course, cause acute toxicity, acute effect, but this is often, not very often. Usually we are dealing with some kind of chronic effects, some sublethal effects which are not noticed immediately, but usually we need some time that we noticed that maybe some impact of one of such compound can result in changing of the ecosystem. Of course, some of the antibiotics, they are very persistent. Some of them can maybe even bioaccumulate or even biomagnify through the food chain. And the significant concern nowadays is due to formation and spread of the resistant genes within different bacterial cultures in the environment, which can cause the unactive mode of the antibiotics during the treatment. We are all aware that these compounds can come into the water, they can come into the soil or in the air, and according to their different physical chemical properties, such as volatility, solubility, miscibility with waters, they can have different transport pathways, they can spread around the environment, and all of these characteristics affects their environmental concentration and finally environmental impact. And we, as the top of the food chain, can be exposed to these compounds through our food, through vegetables, through fruits, through meat products, and of course through the water we are drinking. The groups of antibiotics are very dispersed. We have a large number of different chemical structures with different chemical characteristics, which of course affects their behavior in the environment. As I said before, we are mainly exposed to them and to the products of their degradation in the environment or transformation within the body of the patient, if we are talking about humans or animals. And they can come into our body through drinking water because they can be found in surface or underground water. Where do they come from? Soil. And they can be used in agriculture. They can be also, of course, used in production of animals or directly through some kind of solid waste disposal. And of course, they can come also through municipal wastewater treatment plant because they are not removed there. They are a result of human metabolic products and of course, different pharmaceuticals as well as also the large group of problematic compounds which are found in personal care products. And of course, also due to industrial wastewaters, which can also pass the system and end in drinking water. At the same time, we are exposed to them through food, through different packaging material, from, of course, agriculture and land farming, and as I mentioned before, animal products. So all of this affects their final concentration and then their final environmental impact. So because they are not removed efficiently through conventional wastewater treatment plants, lately we are discussing different methods how to efficiently remove them. So actually the main idea is how to upgrade the existing treatment systems to remove also pharmaceuticals and antibiotics as important representatives of pharmaceuticals. So advanced oxidation processes have lately been well researched and even practically applied for the removal of persistent natural and man-made organic pollutants and in drinking waters and of course also in the wastewaters. They form chemical species in water matrix which are highly reactive and non-selective, and the main component of this species is so-called hydroxyl radical, which is considered to be one of the strongest technically applicable oxidant in water treatment with the oxidation potential of 2.81 volts. They are mainly generated by the combination of oxidant like ozone and or hydrogen peroxide and maybe some catalysts and sometimes in the combination of the source of energy. So usually lately we combine those processes to increase treatment efficiency and of course to reduce materials and energy inputs, to obtain synergistic effects of catalysts, radiation, and oxidants to increase the performance of such process. So as I said before, the main idea of the advanced oxidation processes is to convert problematic organic compound into something which is harmless. That means from carbon dioxide, from hydrogen water, and from different heteroatoms present in the organic compounds, we would like to do some very simple acids, which can be then easily neutralized and of course also return into the environment to go again into the food chain. So as I mentioned before, hydroxy radical has very high oxidation potential. Of course, the oxidation potential of the fluorine is much higher, but however, it cannot be used. And as I said before, it can be performed with a combination of different chemicals. So usually we are talking about homogeneous or heterogeneous processes, where with homogeneous processes, we have all the components in the same phase. With heterogeneous, we have, for example, the catalyst in solid phase and all other parameters in the liquid phase. And in the homogeneous processes, we can use energy or we can have without energy. So we can combine ozone with UV light, with peroxide and UV light, or ozone and peroxide with UV light, or we can also add the iron as the catalyst. And then we can have sonochemic systems with ultrasound energy or application of electric energy. And as a result, we can, of course, increase the performance of the process. In our lab, we are mainly dealing with ozonation because the potential of ozone is quite high because it cannot usually form any solid waste. So according to the physicochemical characteristics of the media, according to the presence, the hardness, the temperature, and the type of the organic, we can reform and force the formation of different oxidative species. So here you have a list, a part of the list of the possible reactions. And the formed radicals can be used for the degradation of the organics. Ozonation can be also increased to improve its performance with addition of the hydrogen peroxide, because it can enhance the formation of hydroxy radical in the combination with the ozone, but its amount and the ratio between the hydrogen peroxide and ozone must be very well balanced, because if we add too much hydrogen peroxide, it can react with hydroxy radicals and actually slow down the reaction. And to avoid such problem, we usually use larger reactor and we add the hydrogen peroxide at several points, or use different cascade of the reactors to enhance the hybrid potential of these two chemicals. And of course, we can also increase the performance of the oxidation processes by addition of the UV light, which enhance the formation of hydrogen peroxide from the ozone. And then you have the combination of the hydrogen peroxide and ozone, as mentioned in the previous system. So we actually form the hydrogen peroxide in situ in the reactor, and then we start the set of the reaction as mentioned before. And another option is addition of the catalysts like iron 2+ or other metals with comparable characteristics. And they, together with hydrogen peroxide, can also form hydroxy radicals, which can then react and start the degradation of the organic compound. Here again, the concentration and the amount of the iron and hydrogen peroxide is also very important, because if we add too much hydrogen peroxide, then it can react with hydroxy radicals and again reduce the rate of the reaction. So to give you a little bit impression how we can compare these processes, I would like to present you a case study with thiamulin, which is, as I said before, antibiotic which is used in veterinary medicine for treatment of the pigs and hens and chickens, so in the poultry. And it is quite toxic to the microorganisms of the activated sludge. So that means that if it comes into the biological wastewater treatment plant, it can, due to its persistency, reduce its performance. So it's maybe good to partially degrade it or remove it prior to the biological treatment. So as you can see here, where the toxicity is presented in this table, it can, in concentration of 1,001 gram per liter, it can reduce the performance of the microorganisms of the activated sludge for 50%. And the main problem is also because it quite strongly inhibits the nitrification process, so degradation of ammonia into the nitrate, which is very important in every biological wastewater treatment plant. So there is a reason to remove it, to use a process for removal from the wastewater and not let it go to the biological treatment, or even, of course, it cannot be spread into the environment. And we can also see here on this graph that it is not very good biodegradable, because at non-toxic concentration of 100 milligrams per liter, it degraded in almost 44 days, only 40%. So that means that it would be persistent in the environment. So we have run very different experiments. We have ozonation at increased pH, ozonation with hydrogen peroxide, with addition of iron, with combination of iron and hydrogen peroxide, and then with iron and addition of UV light, and of course also the combination of ozonation, hydrogen peroxide, iron, and UV black light. here are just the parameters of the system of our reactor. Because we have different reactors, these experiments presented here were done in a lab reactor of 300 milliliters, but of course we also perform experiments in larger reactors. Before the experiments, we also test the thermal stability of the compounds, that we are sure that any processes applied is the results of any process applied are really the result of the chemical reactions in the process, not some kind of changes of the temperature. And we can clearly see that there is actually no changes with the increased temperature in the concentration of the thiamulin. And here in this graph, we can see removal efficiency according to the chemical oxygen demand, and we can see that more combined process with different chemicals, more efficient it is, and actually the most efficient is combination of iron, ozone, and hydrogen peroxide, as well as UV light, where we attained almost 95% of the removal. But however, this process is of course the most expensive one, because you use the most different chemicals, you use the energy, and with the measurement of TOC, total organic carbon, we confirm that it is not complete mineralization. So we see that the degradation was only partial, because if the degradation was complete, that the thiamulin completely converted to carbon dioxide and water, these values would be the same, but we see that they're a little bit lower. So it can be clearly seen that those processes are effective, that ozonation can be enhanced with addition of different chemicals and oxidant and energy, but however, we have to always consider the price of such processes. And but on the other hand, we can see that toxicity was at the beginning, for example, for this concentration 44 of this thiamulin solution, and it was even increased in some processes. That means that formed metabolites, formed byproducts can be even more toxic than initial compound of the thiamulin. But however, in the last, where we have the combination of all processes, the toxicity was significantly reduced. And on this upper graph, we see that biodegradability was actually not increased, and this confirmed that different products were formed which are more or less the same toxic. They are not more biodegradable, and we try to identify them. And here is just the part of different compounds which were formed during those processes. And we can see then that in some cases, thiamulin did not degrade during ozonation, but it just formed different fragments. So this is the reason that toxicity was still present, that biodegradability was not increased, and we can conclude that this process is actually not so efficient as seems at the beginning. So to conclude, I would like to say that, of course, antibiotics present a significant risk to the environment, because we can find them almost everywhere in our environment. Many of them are quite persistent. Some of them can always have bioaccumulation potential, so we are not aware what is actually their long-term effect. Oxidative degradation of selected antibiotics and their mixture and wastewaters containing antibiotics was proven to be efficient in many cases, but however, it depends upon the structure, the concentration of the antibiotics, as well as the process selected. And it has to be optimized for each compound separately, and we have to decide where to put some additional process for the treatment before biological process, or just before the outlet of these compounds into the environment. So there is always a need for some kind of laboratory and pilot testing, and we have to consider a lot of different parameters before we can make the final decision where to upgrade the existing treatment plant to reduce the release of such problematic compounds, micropollutants, into the environment. And that, of course, brings me to the end of my presentation. Thank you very much again for your listening. And if, of course, we have time, I would be very glad to answer some of your questions. So please, thank you. So I cannot hear, so there is no questions.
With that, the conventional techniques cannot remove antibiotic contaminated in our domestic wastewater treatment plant or in environment. And in Thailand, we have also the same problem in Europe and in so many countries that we got a lot of the contaminated area, especially in aquatic farm, because some of the farmers use antibiotic to prevent their production. And my question is, because according to your presentation, you use the advanced oxygen process such as the ozone with some of the hydrogen peroxide or some of the oxidant to generate the free radical, for example, hydroxyl radical or sulfate radical, including the UV reactor. And do you have I have two questions. The first is, can you suggest us that if we really want to use the advanced oxygen process for a Thai farmer in their farm, do you have any idea which one is the cheapest for the lowest cost? Because the ozone is a high cost electricity.
Yeah, actually, I would say the cheapest one is maybe something with iron and hydrogen peroxide. So that means the phantom oxidation, because usually the iron 2+ is cheap, or you can use maybe some crushed waste metal. If you have some metal waste, or you have maybe a soil with a lot of iron, and then you can add peroxide, and you can have the column where you can try to treat such water. But it depends upon the type of the antibiotic. But I would say that hydrogen peroxide is the cheapest. Maybe you are efficient only with hydrogen peroxide, or maybe you can combine with iron 2+, or even iron 3+ if you have some waste. But these processes are usually not so efficient because iron 3+ has to be converted first into iron 2+ that it became active. So it depends.
Yeah, thank you for your answer my question. And my second question is, according to your presentation about the removal of tambourine, and you test with the toxicity, and you conclude that when you using the ozone with the ion and hydrogen peroxide and UVA, you get the best result for inhibit of the microorganism just 5%. I'm not quite sure why this combination is the best for toxicity inhibition for microorganism.
Yeah, you get the lowest inhibition because actually I didn't present results here, but after these processes, you get only small fragments of organic compounds, and it seems to be that remained fragments are very well oxidized. So you have a lot of acidic groups on these remained rings, and these are groups which are not very toxic. So actually the products are the less toxic, and this as a result caused the lowest toxicity of this mixture after the advanced oxidation process.
Thank you. Is there any question from floor? Yeah, one question from Associate Professor Dr. Shanat.
Hi, can you hear me?
Very, very low, very low.
I have a quick questions about the any antibiotics or any pharmaceutical products that you want to be treated with your system or your oxidative systems, because some of it can just only be soluble in lipid, right? So how can you work with that? Because I would say that your system, your oxidative system, will only work with the aqueous solution.
Yeah, those compounds, if they are treated in such a way, they have to be at least, they have to be water soluble. But if they are not water soluble, of course, if they're treated if they're dissolved in the lipids, you cannot treat them because you will use enormous amount of the oxidants for the treatment of the lipid phase and everything. So no, it's just for those which are soluble in the water, and as a result, they are very mobile in the environment, because the compounds which are not very well water soluble, they're usually not so mobile in the environment, and maybe they are not so problematic from this point of view.
All right. Thank you so much.
Thank you.
Okay. Thank you so much, Professor Andrija. I think we run out of time right now, and we are going to move to the next speaker. Thank you so much for your help.
Thank you very much. Bye bye.
Bye bye. And for the next speaker, it is Professor Dr. Patrick Brotikam from Center for Energy and Environmental Chemistry, Jena, Germany. He's going to present his topic in the degradation of organic micropollutants in water by cavitation and cavitation assist advanced oxidation processes. And before Dr. Patrick give his presentation, let me give you for his background. Dr. Patrick Brotikam is head of the research group Water Technology, Cavitation, Renewable Resources at the Center of Energy and Environmental Chemistry, or CEEC Jena, at the Friedrich Schiller University, Jena, Germany. And he also is a head of research group Reaction Technology Water at Fraunhofer IKTS. His research is focusing on anthropogenic micropollutants, pharmaceutical microplastic, and he also conduct with the new techniques for water treatment, especially advanced oxidation process, membrane adsorption, and cavitation. In the cavitation field, his research focus on the generation of cavitation hydrodynamic, acoustic, combined the analysis of cavitation field, cavitation reactor design, and use of cavitation in different applications, water treatment, biodiesel synthesis, and modification of surface. Right now, ladies and gentlemen, I give you Dr. Patrick Brotikam from Jena, Germany, and the floor is yours.
Thank you. I will try to share my slides. Hopefully you can see it now. So can you see the slides or a presentation mode?
Yes, we see your slide. Yes.
Okay, presentation mode or the whole slide?
You have to go to presentation view.
Okay, thank you. So now it should work, right? Okay, I hope it works. Okay, if not, you can give me a hint. Yes, thank you for the introduction and for give me the chance to present some of our research. So we are a bigger group in Germany which are interested in advanced oxidation process, and the presentation before give a very broad overview of what techniques you can use. But I think nearly newer technique you can use is the cavitation processes or combine cavitation processes with well-known oxidation processes, and that's the reason I want to introduce you to that topic. So first of all, since I think not everyone is familiar with cavitation, I want to give a short definition. Cavitation, to say with the words of Ken Zaslik, which is the founder of that technology, is the formation, the growth, and the implosive collapse of bubbles in liquids. So what we are focusing on is small bubbles which are collapsing. So I bring you here a video. This is a high-speed video from a collapsing bubble. You can see the bubble expands and then the bubble shrink. So this is an implosive collapse. And the size of such a bubble is in a region of some micrometers, and the collapse, the time in which the collapse takes place is all a small amount of milliseconds. So the process is very fast, and the volume contraction has a factor of more than 10,000. So you have a very high volume contraction in a very short time. So you produce very high temperatures and pressures in a very small area. So when you have it in water, you have millions of that small bubbles that are collapsing in parallel. So yes, this is the cavitation process we try to use for degradation of organic micropollutants in water. So what effects are connected with that aspects? We have on the one side, we have chemical effects. These effects originate from that hotspots, that high temperature and pressures within that collapsing phase. So when you have that high pressure, water molecules can undergo the cleavage of that bonds. So you can form out of water OH radicals. You can also form perhydroxide radicals, hydrogen peroxide, some oxidative species. And these species are able to degrade such compounds like bisphenol A, for example, here it's shown. These are the chemical effects. On the other hand, you have physical effects. They are also due to the asymmetric or symmetric collapse of that bubbles. So you can produce microjets, hydrodynamic shear forces, shock waves, microstreaming, something like that is all directly connected to a very fast movement of the bubble. So these effects you can use for enhancing mass transfer to surfaces, to cleaning surfaces, to mixture something. You can use it in different applications. So yes, these are the principal effects are directly connected to that collapsing bubble. Okay. Slide. Yes, the next question is how we can produce or generate cavitation bubbles. So I would say mostly common used technique is acoustic cavitation. That means ultrasound, and you have an acoustic wave, and that wave leads to an expansion and compression phases, and bubbles are growing in a time frame, and they reach a critical size and then collapse and show that hotspots, that high temperatures and pressures. And it depends a little bit which frequency you use. If you use a low frequency ultrasound, that means in a region of 20 to 40 kilohertz, you produce more or less a physical effect. So it's more mass transfer mixing. If you use higher frequencies above 100 kilohertz in a region, you have more that sonochemical effect. That means more OH radicals, for example. And the reason is that the time frame of the collapse becomes smaller and smaller if you increase the frequency. So the time step of a collapse is much faster, and so the temperatures and pressures you can reach are also faster, leading to higher concentration of OH radicals. The other, or something newer technology used in water treatment is hydrodynamic cavitation. This is a flow cavitation. You use, for example, pipe flows and increase the fluid velocity in a system. And due to Bernoulli equation, that says that the sum of static and dynamic pressure is a constant, you can easily see if you increase the fluid velocity, the static pressure decrease. And if you decrease the pressure, you can form cavitation bubbles. And after the pressure recovers, the collapse takes place here. And you can generate this very easy in a pipe flow and introduce something like an orifice in a system or nozzle or something like that. But we also use specialized restrictions here to use. But these are in general these two methods to generate cavitation for processes. There are lots of applications you can use cavitation for. In this presentation, I really will focus on water treatment and use it at advanced oxidation process, but you can also use it for disintegration, for biogas, for mixing biodiesel, disinfection, synthesis of amorphous nanoparticles, surface modification, cleaning, sonochemistry, something like that. The list is very long, and you can find lots of applications of cavitation processes in the literature, but there are some problems occur concerning the industrial usage. One is the high energy demand, and the other is the scale up. That's the main reasons, one of the main reasons why you can find it in the industry up to now. We are trying to get better technologies to to face these problems and to find solutions for that. One is new cavitation techniques, namely hydrodynamic cavitation and a combined system, which we are calling HAC, hydrodynamic acoustic cavitation. And on the other hand, the combination of processes, the photocatalysis, electrochemistry, or ozonation, for example. And one thing I want to show you is hydrodynamic cavitation. This is a flow cavitation system. And first of all, we want to understand how it works, what parameters are affecting the degradation of compounds, for example. And you can see here is some pictures of this is a pipe, and at the end of the pipe there is an orifice, and we have here different inlet pressures starting from 15 to 30 bars, and you can see that you have a bubble field. And the bubble field expands if you increase the pressure. That's clear. So what we are looking at are the bubble field itself, the bubble size distribution, and we correlate this with the rate constant, for example, here for the degradation of bisphenol A. And you can see there's a linear regression concerning the inlet pressure and the rate constant for for the degradation of bisphenol A. On the other hand, we use also luminol reaction, which you can see here, which luminol reacts with an OH radical and form a blue light. And we can see where in the reactor or where in the bubble field OH radicals in which quantity is produced. And we can see it here that the blue light expands since the bubble expands, and you can find it nearly in the at the beginning of that nozzle or that orifice. But you can see if you increase the pressure, you also increase the production of OH radicals, you increase the degradation of that micropollutant of that bisphenol A. As I mentioned before, the HAC concept. When you take a look in the reaction of cavitation, you have two things you have to be in mind. One is the number of bubbles. This is an important factor since this is directly connected to the reaction volume. The other thing is the collapse intensity, and this is more or less related to the reaction conditions. So if you are interested in the conversion, for example, water or the production of OH radicals, you need a high number of bubbles which are collapsing at very high intensity. That's should be the best for production of OH radicals. So having a look at the single processes, that means acoustic cavitation and hydrodynamic cavitation. When we have a look at the bubble density, this is very low with acoustic cavitation, but the intensity of a collapsing bubble is very high. In hydrodynamic cavitation, the bubble density is very high, but the collapse intensity is low. So that leads us to the idea or the thesis that we combine this first the hydrodynamic cavitation, which is producing a high bubble density, and afterwards the acoustic cavitation, leading to a high collapse intensity. And we hope then if we combine these two, we get very high bubble densities, collapse intensities, a better energy efficiency, and better scale-up opportunities. And here are the main result of that investigation. We construct a reactor concerning of hydrodynamic cavitation. This is here in the beginning, an orifice, and the flow goes into reactor, and here on the left and right hand side out. And we have here the orifice, and here the sonotrode for ultrasound. And you can see here is the bubble cloud. And when we have a look more in this region, you can see it here. It's a picture where the black points are bubbles. And you can see here, this is the pipe flow with the orifice seen here. And you can see the black one is the bubbles and the blue one is the luminol. This is the part of the bubbles which are active, which are producing OH radicals. And here, this is the sonotrode, which are vibrating in that way. So when we measure the luminol and we give it in a black-white manner, you can see this is the hydrodynamic cavitation. You can see it here. This is the luminol field for the ultrasound here from the sonotrode, and that one is the luminol picture from the combined system. When we overlay it with the single methods, you can see there is additional black points, which are leading to a big synergy of that both techniques. So when we combine it, we get lots of more OH radicals by combining these two processes in the right way. So we also want to show it by the degradation of a micropollutant. We take here carbamazepine as a pharmaceutical, and you can see if you have that single methods, it takes nearly half an hour to come down to 50%. It's a half-life time. And with a combined system, you come down to 3 minutes, nearly 3 minutes to degrade 50% of the initial concentration. And that's the reason for that is we combine these two processes, we get more energy efficient with that technique. But we also combine cavitation processes with other processes. One is the photocatalysis. The photocatalysis itself has a very low energy consumption. It's more related to hydrophilic pollutants due to the surface structure. You have a mass transfer limitation and have sometimes problem with fouling and scaling in that system. Cavitation on the other side has a comparable high energy demand, but can degrade more hydrophobic or volatile compounds, shows a high mixing, and is able to clean or deagglomerate that catalysts. So when we combine these, we hope that we can enhance the mass transfer, so we get faster kinetics. We can clean the surface for anti-fouling, anti-scaling effects, and we can degrade hydrophilic as hydrophobic pollutants in a high with high kinetic constants. So here I show you some results of our research. So we construct a reactor consisting of ultrasound. Okay, it's in German. This is an ultrasound, and this is a panel for UV LEDs. And here we have plates with titanium dioxide, and we can adjust the distances and lots of other parameters. Here you can see a photo. This is a bigger reactor. It consists of, I would say, 10 liters around. And here you can see the results from the degradation of naproxen, which we are using as model compound here. You can see the normal photocatalysis takes nearly 3 hours to degrade 80% of the initial concentration. When you do this with sonophotocatalysis, you need not more than 20 minutes. So you get a really faster process. And concerning the energy demand, you need 10 or 20% of the original energy of a photocatalysis. So it's faster and it's cheaper. So it's very interesting technology. Another thing is sonoelectrocatalysis, where we use boron-doped diamond electrodes. We have nearly the same problems here, since this is a surface and mass transport limited reaction. You can introduce cavitation within, and we also found enhanced mass transfer, disruption of diffusion in Helmholtz layers, and surface cleaning. In electrochemistry, also the removing of gas bubbles on the surface of that electrodes are important factor there. You can see here it's a scheme of a reactor. You have a high frequency system, a low frequency sonotrode, the two electrodes. You can hear a picture. And this is the result concerning bisphenol A degradation for electrochemistry, sonochemistry, and sonoelectrochemistry. You can also find here a synergy, and the system has to be improved. We hope that we can see similar effects like in the sonophotocatalysis. We also use that technique for sensors for the detection of COD, for example. Next slide is the combination of ultrasound with ozonation. We try also to do this because ozonation has a high rate of oxidation species, but has also mass transfer limitations and a comparable low mineralization rate. If we combine this, we hope that we can enhance the mass transfer and decompose oxygen in the cavitation bubble, since that gas can come into a bubble. And we think that the ozone can be activated somehow, because we leading in this process to an oxygen radical, which then reacts with water to form more OH radicals out of one ozone molecule. So, and if we do this, here is a reactor. You can see this the inlet. It's ozone inlet. This is, for example, an ultrasound, and we have two outlets in the system. And you can see here is, okay, German, sorry for that. This is concentration of benzotriazole as a model compound, and this is reaction time. And you can see here the single methods needing more than 30 minutes, and you come down to 4 minutes. So it's the same way. You are much faster and much more energy efficient. And the last slide I have for you here is the use of machine learning for the prediction of the rate constant in AOP processes. So what we are doing is we want to predict out of the molecular structure the reaction rate in different AOP processes. For example, ultrasound, photocatalysis, hydrogen peroxide, electrochemistry, all of that methods. And we have lots of micropollutants, and no one knows which pollutant is the best or which method is the best for a specific pollutant. And with the use of machine learning, we can predict. And we start here with ultrasound. And you can see here, this is an experimental K, rate constant to the predicted. And you can see we are nearly together. So the model works very well. So this is also a very interesting technique for the future, because you can use in silico methods to predict structures and methods and bring it together. And this is my last slide. I want to thank my group, our partners, our funding agencies, and you for your attention. Thank you. Sorry, I was a little bit long, right?
Sorry for the technical problem. Thank you so much for your talking from very interested in the topic of degradation of organic micropollutant in water by cultivation from Dr. Patrick from Jena, Germany. I have a quick question that because the cavitation technique can generate hydroxyl-free radical, and when we compare with the other advanced oxidation techniques, when we compare, for example, with the removal of bisphenol A contaminated in water, how long does it's gonna take when we compare with the other technique? For the cavitation technique is faster than the other advanced oxidation techniques or not? I'm not quite sure that which one is the more powerful.
Mm-hmm. So from my point of view, the cavitation as a single method is not useful for AOP. So you can use it only in a combination where you find very interesting methods. If you use only ultrasound or hydrodynamic cavitation and want to test it against photocatalysis ozonation, the processes are too energy inefficient. So you need more. You can degrade the pollutants, but it takes too much energy. But if you combine it, the combined processes are much faster and energy efficient than combining with comparing with photocatalysis or ozonation. So the idea is to combine it and not to use the single methods. The single methods are not useful for AOPs. They are useful for other techniques, but not for AOPs.
Yeah, thank you so much for your answering my question. And I have a quick question that if I use some of the electrochemical techniques that I saw some your slide that show the cavitation technique can remove on of the some of the particle on the surface of the metal. If we combine with the electro-oxidant method, it would be better, right? I'm not quite sure. If we want to increase the efficiency of the electrochemical oxidation, can we combine with the cavitation method?
Yes. We combine it. So we use it for we use boron doped diamond. So this is not so I think the problem is cavitation erosion. So you can treat or destroy surfaces, but it's a question of distance and energy. You can balance it to have a stable surface, and you can also increase electrochemical oxidation processes. Yes, it works, definitely.
Thank you. One more question from Associate Professor Dr. Chanat.
Yes.
Hello. I have just one question.
Yes.
General rule of thumb when can you hear me now?
Yes, better.
Okay. General rule of thumb of when we want to use two things like two oxidants or two oxidation process together is that it has to be well balanced, right? I mean, between, like, for example, ozone or hydrogen peroxide concentration, those kind of things. So if we want to use cavitation technology with some other oxidative methods, how do we control this cavitation? Can it be measurable? I mean, how do you do it?
Mm-hmm. Okay, so it depends on which process you want to increase or want to optimize. So if you have, for example, surface processes for like photocatalysis or electrochemical oxidation, you have always a surface in which the reaction takes place. You produce OH radicals near to a surface, and you have to bring the pollutant next to the OH radical. So then you need a system for cavitation that only enhance mass transfer. You don't need that much energy. It's something like a UV light. It's very low. But if you, for example, use it for the combination of ozonation, you need a little bit more energy because for mixing mass transfer in the system and activate the ozone. So you can combine it, and it depends on the frequency and the energy and the distance, and you can measure the effects by, like, for example, hydrophone. You measure the energy of a sound wave depending on the distance. There are some techniques you can measure. It's called cavitation measure value, or something like sound wave characteristics. But you can also use that luminol, where you can get an impression where in the reactor the OH radicals can produce. So there are some methods you use and put it together to get an understanding how it works, and you can control it via energy input. It works very well.
Thank you so much. Very interesting.
Thank you.
Thank you so much, Professor Patrick. And right now we are going to move to next speaker. Thank you so much for your help for our seminar. And for the next speaker will be Professor Dr. Yvonne Stanker from Department of Parasitology, Mycology Center, Hospital University of Montpellier, France. He is going to talk about the state of art of molecular and cellular biology to study the core cellular processes of the divergent eukaryote leishmania species. Before he is giving a seminar, let me give you for his background. Professor Dr. Yvonne Stanker was born in 1971. He was first educated as a medical doctor specialized in clinical hematology in the University of Paris, and then obtained a PhD at the Pasteur Institute under the supervision of Professor Arthur Schwepp on the physiopathology of the severe form of Plasmodium falciparum infection. In 2006, Professor Yvonne moved to Montpellier, where he has been tenured as associate professor in 2010, and he earned full professor in 2021. As an MD, Professor Yvonne is responsible for the molecular diagnosis of parasitology and mycology infection at the University Hospital Center of Montpellier, which include molecular identification and NGS approaches. Finally, Professor Yvonne teaches parasitology at the Faculty of Medicine at the Faculty of Science of Montpellier and is responsible for the PhD program of the LABEC Paragraph. Today, Professor Yvonne will present his topic in the state of art of molecular and cellular biology to study the core cellular processes of the divergent eukaryote leishmania. And right now, I give you Professor Yvonne. The floor is yours.
Thank you very much. Thank you for giving the opportunity to present my work, and thank you for the nice introduction. So I can't move the slide. So yes, I mainly work on leishmania and also on Trypanosoma brucei. Both are trypanosomatids that belong to the order of kinetoplastids. Here I present the complex life cycle of leishmaniasis. I won't go into detail. Just leishmania leishmaniasis are responsible for leishmaniasis, a group of mainly zoonotic disease transmitted to humans and animals by the bite of phlebotomine sandfly. It corresponds to the third vector-borne disease after malaria and lymphatic filariasis. Leishmaniasis occurs in around 100 countries all over the world, and the clinical range from self-healing cutaneous ulcer to the fatal visceral leishmaniasis. Here I present the tree of life of the eukaryote presented by Baldorf. This is just to highlight that the kinetoplastids are divergent eukaryotes, far from the model organisms of human Drosophila and yeast, but also far from other parasites such as the Acomplexa, Plasmodium, and T. gondii. As divergent eukaryotes, Leishmania has many original features as compared to user eukaryotes. For example, the mitochondrial DNA, also named kinetoplast, which has given the name of the order, is made of thousands of interlocked DNA circle, mini and maxi circle. Genome organization is also original with long polycistronic transcript units made of non-functionally related genes. There is almost no RNA polymerase II, and the regulation is mostly post-translational. Genes doesn't have any introns, but there is an intensive transplasing of a splicing leader to the 5' end of all messenger RNA. The physical elements of the mitotic cycle are also original. Mitosis is closed, which means that the nuclear envelope remains intact all over the cell cycle. The nucleus and kinetoplast cycle are synchronized, but shifted and independent. Independent because when kinetoplast cycle is blocked, cytokinesis is blocked, but mitosis occurs, and we can observe multinucleated cells. Conversely, when the nuclear mitosis is blocked, cytokinesis occurs with the production of anucleated cells, also named zoids. Extensive bioinformatic analysis failed to identify extensive bioinformatic analysis failed to identify any kinetochore proteins, and ultrastructural studies detected electron-dense plaques. But there is a high inadequation between the number of kinetochores and the number of chromosomes homologs. There is 8 electron-dense plaques or kinetochores for 11 megaseries chromosomes for TBCI, and only 6 for the 36 chromosomes of Leishmania major. Until 214, and the work made by Bungo Akiyoshi in Kiss Girl Labs, no kinetochore proteins were identified. They have used a screen based on the localization of protein of interest. The criteria were that protein of interest should be expressed late in the cell cycle, and that their localization was made with dots appearing in the nucleus around the ACE phase and moving to the opposite poles during anaphase. With the protein, the tagged protein of interest, the candidate protein of interest, they have performed ChIP-seq in order to enrich at megabase chromosome centromeres. And with the good hits, they have done serial immunoprecipitations and mass spec identification. They were able to obtain 20 and more recently 25 unconventional kinetochore proteins in kinetoplastids. We took advantage of the high orthology between the genomes of Leishmania major and T. brucei, and we have used the orthologue of TB KKT1 in Leishmania to perform ChIP-seq. We first checked that the localization of LM KKT1 GFP was compatible with that of a kinetochore protein. And then we use it as a probe for ChIP-seq. And LM KKT1 GFP ChIP-seq shows a single ChIP-seq peaks corresponding to the 36 centromeres, one per chromosomes. So in this article published in EMBO reports, we were able to show that LM KKT1 display kinetochore-like dynamics of intranuclear localization throughout the cell cycle, that LM KKT1 shows single narrow peaks per chromosome that correspond to the centromeres. And concerning the Leishmania centromeres, we could conclude that they are essentially non-repetitive and display great interchromosomal sequence heterogeneity. They are also characterized, most of them are characterized by two conserved motifs and a higher density of retroposons as compared to the rest of the genome. One of the most original features concerns ploidy and genomic plasticity. The ploidy of the divergent protozoan parasite Leishmania was controversial, and the debate was fed with the polemics between sexuality and clonality. When I arrived in Montpellier 2006, we were able to demonstrate that by fish experiments that Leishmania is aneuploid, in that sense that every chromosome can be mono, di, or trisomic, but also mosaic in the sense that all cells in a population do not possess the same number of copies of a given chromosome. These findings has been observed in several species. In fact, in all the Leishmania species assessed so far. It was confirmed by WGS data at the population level, and also very recently in a collaborative work led by Jean-Claude Dujardin in Antwerp by single-cell sequencing. The biological and genetic consequences of this mosaic aneuploidy has been discussed by our group and in others in several published works. Here, I want to go in more details in the molecular moleculisms which underlies mosaic aneuploidy. By fish experiment, when we focus on dividing cells, we have noticed that we observed asymmetric chromosomal allotments with an odd number, either 1 and 2, with an odd total number of chromosome homologues, 1 and 2, or 2 and 3. This feature is distinct for asymmetric chromosomal allotment with an even number, 1 and 3, which is observed in segregation defect. So this asymmetrical chromosome allotment with an odd number, in our point of view, suggests a replication defect. And we were able to propose a model which involved double firing and non-firing events of origins, leading to monosomic and trisomic cells. To determine the DNA replication dynamics, we set up the DNA molecular combing to these parasites. Briefly, it's based on sequential labeling of asynchronous cell population with modified nucleotides. Then we embed the genomic DNA in low melting agarose plug, and we comb the DNA with a uniform extension of DNA fiber on a selenized cover slip. We detect IDU, CLDU, and DNA with antibodies, and we are able to determine the replication parameters, replication fork, velocity, and speed, and to measure anti-origin distance and to estimate the number of origins per genome. This data was published in scientific report. Very briefly, we were able to show that Leishmania, as compared to T. brucei and higher eukaryotes, has large IODs of around 200 kbs, high speed. It is even the highest speed ever recorded for a eukaryote. But we were not able to show any evidence for double firing events. We are now setting up the CNS technique in order to finally map the origins. This is based on the purification of the Ozaki fragment on sucrose gradient to prepare a library and perform next generation sequencing. Analysis, the bioinformatic analysis, and the replicates are ongoing today. Now, I would like to present you the strategy we use based on cell biology data for the gene of interest we studied. We first localized the protein of interest, and for years we were using episomal expression of a tag protein. We also generate mutants. Here we have to face that generating knockout were very difficult and most often impossible in Leishmania, and that most of Leishmania species lack the RNAi pathway. This explains that many studies were performed on T. brucei, taking advantage of which process the RNAi and taking advantage of the high synteny shared between these two parasites. However, since 2015, we use CRISPR-Cas9 strategies. Briefly, a short notice on CRISPR-Cas9. It is based on double-strand break at a specific site and on the replication by homologous recombination. The three actors are Cas9, the sgRNA, and donor DNA. Cas9 can be produced by episomal expression or in vitro. sgRNA and DNA can be episomal expressed and PCR based. We were the first to set up the technique to Leishmania, but our technique has been supplanted by that made by the Everglades lamb, which is PCR based and much more efficient. However, we were able to propose an inducible system combining the CRISPR-Cas9 and dicray system. Here I want to present our ongoing lab work on the role of nucleopores and nucleoporins in the maintenance of the ploidy. Briefly, the nucleopores are large multimolecular complexes. They have canonical functions, means the transport of molecules, and a large range of non-canonical functions. We focused on MLP1 and MLP2. As compared MLP2, we and others has published article that shows that MLP2 has pleomorphic roles in several core biological processes, in particular in chromosomal distribution and also in chromosomal segregation. Concerning MLP1, using CRISPR-Cas9 to tag the protein, using immunofluorescence assay and co-labeling, using super resolution microscopy and immunoelectron microscopy, we were able to show that MLP1 localizes at the nuclear pore basket. All the attempts to generate a null mutant with all the CRISPR-Cas system published failed. We have to develop the conditional deletion system based on the introduction of LOXP surrounding the gene of interest by the CRISPR-Cas and to induce the edition. We were able to induce efficient gene deletion and protein depletion with this system. And the mutant parasite presents a severe growth defect. After inductions, the mutant parasite shows an abnormal nuclear and kineto pattern. There was a significant decrease in 1N, 1K percentage of 1K, 1N cells, and a significant increase in abnormal cell types, in particular, cell with nuclear envelope displaying abnormal extension. In order to look if MLP1 affect the nuclear envelope and the nuclear pore complex structures, we perform collaborating studies with other NUPs, either in the cytoplasmic or in the middle or in the nuclear side of the pore, and also with a laminin. And by immunofluorescence assay and with super resolution microscopy, we were able to show that MLP1 deletion severely alters the nuclear morphology and the nuclear envelopes. We also look at the RNA transport, which is one of the role of the nuclear pores, and we were able to show that RNA transport was altered in MLP1 depleted parasites. As concerned the ploidy was also modified. When we look by fish, the zomi of chromosome 8, which is mostly disomic in the parental side, we see a large decrease in disomic cells in favor on monosomic, trisomic cells. When we look at the chromosome 2, which is mainly monosomic and disomic, we also observe a modification of the equilibrium, but this case toward tetrasomic and even more than pentasomic cells. Since the ploidy was modified, we decided to look what was going on the kinetocores and looking for KKT2. We could observe that the distribution of KKT2 was perturbed and modified during mitosis in the mutant parasite. We also look at the mitotic spindle. To do so, we make a colocalization with a spindle associated of an kinesin F. And in the mutant parasite, we see that the localization of kins was highly altered. So to conclude, we could say that the MLP1, what we observed in leishmania with MLP1 was also confirmed in the sister parasite T. brucei, and that MLP1 is a nuclear basket protein essential for cell survival, essential for nuclear envelope and nuclear pore complex integrity. It is involved in cell cycle progression and DNA contents. It plays a role in KKT distribution and mitotic spindle formation. The role on ploidy is slightly different between T. brucei, where it is implicated in the diploidy maintenance and symmetric divisions. In leishmania, the role looks more like in genome integrity and genome and chromosome stability. But in both cases, MLP1 is implicated in the maintenance of the ploidy pattern of the T. brucei or leishmania parasites. This work has been done in Montpellier, southern France. And my team that I acknowledge here developed 3 axes. One axis, a large axis on the basic cell process, and I present you the data obtained in this axis. But we also have genetics, genomics, and molecular epidemiology on Leishmania and Babesia. And this axe collaborates with Anamika from University of Calzart. And as MDs, we also develop bioclinical and translational research in frame of the molecular biology pole and the National Reference Center of Toxoplasmosis, the National Reference Center of Leishmaniasis, and we also have access in mycology. I thank you very much, and I'm open for questions.
Is there any question from the floor?
Yes, I have one question. So, okay, in your opinion, for now, and all the molecular and cellular biologies of Leishmania we have known now, which is the best methods or technique you will recommend for diagnosis of the Leishmania infection in human and animals?
In both cases, it depends if you are looking for an acute sickness. In that case, molecular biology is the best, and and sampling blood is as good as sampling bone marrow, and the target should be a kinetoplast target because it is much more repeated than a ribosomal target. But you have to do it in a very good lab, very protective, because you are a higher risk of IO contamination due to the large number of kinetoplast target in the genomes. Serology could also be used, but you have to be cautious with if your patient is immunosuppressed. Finally, for the cutaneous form, the best and only approach is PCR or the microscope, but it is less sensitive, of course.
Mm-hmm. Okay, thank you.
Is there any question? I think there is no any question from the floor. Okay. Thank you so much, Dr.
So in the chat box, someone say best presentation indeed.
Thank you very much.
From Dr. Anamika.
Thank you, Anamika.
Okay. On behalf of Faculty of Veterinary Technology, I would like to say thank you to Professor Dr. Yvonne Stange from University of Montpellier, France. And next speaker is Dr. P. Chanakan Chopjit from Faculty of Public Health, Kasetsart University, Thailand. She will give her in the topic is distribution of MCR harboring Escherichia coli confer to non-cholesterol susceptibility isolated from slaughter pigs in Thailand. Before she give her talking, let me give her some background. Dr. P. Shanakan Chopjit obtained her doctoral degree in medical microbiology from Konkan University. She is currently works as a lecturer at Faculty of Public Health, เกษตรศาสตร์ University เฉลิมพระเกียรติสกลนคร Province Campus. Her research works focusing on antimicrobial resistant bacteria in human, animal, and environment. And right now, the floor is yours for Dr. P. Shanakan.
Okay, can you hear me, everybody?
Yes.
Okay. First of all, I will tell you.
Dr. P. Shanakan, could you show the full presentation, please?
Okay. You can see it.
Yes, it
It's
is.
a full, right? Okay.
Yes, it is.
Nowadays, first of all, I will tell you first is our electricity and our internet maybe have some problem right now. Maybe it's not stable. Okay, let's start to my talk today. First, I would like to thank for your inviting to talk in this conference. And let's do my talk today. My topic is the distribution of the MCR harboring Escherichia coli conferred to non-cholesterol susceptibility isolated from slaughter pig in Thailand. Okay. Okay, for my presentation today, I will divide into 4 parts, including the first is the overview of cholesterol antibiotic. Second is the mechanism of cholesterol activity. Third is the epidemiology of cholesterol resistance, and the last topic is our study or our research. For the cholesterol, the cholesterol is the antibiotic agent belong to the polymyxin antibiotic group, and the first isolate was found in Japan into 1949 from Bacillus polymyxin, that is the gram-positive bacteria. And nowadays, polymyxin consists of polymyxin class A, class B, C to E, and polymyxin and cholesterol is the polymyxin E. Right now, the polymyxin B and E was used in the clinical practice, and in the general structure of the polymyxin B and E have the high similarity, but different only in the position number 6 on the polymyxin E. We will see the leucine, but for the polymyxin E, polymyxin B, we will find the phenylalanine. Why the cholesterol is important? Because of this antibiotic is the last resort or last choice for treatment who infect with the carbapenem-resistant enterobacterial. So it means that this antibiotic have the effect agent to the most gram-negative bacteria, and is used in human and veterinary medicine in 1952. For the commercial, nowadays, cholesterol have 2 forms, including cholesterol sulfate for the oral and topical use, and the second is the cholesterol methasulfonate for the parenteral use. Let's do the mechanism of cholesterol activity. This antibiotic will disrupt the negative charge of outer membrane of the gram-negative bacteria, lead to permeability change and leaking of the cytoplasmic content into the cells, and finally led to the cell death. However, after the colicin were used in the worldwide, and few years later, the colicin-resistant bacteria will occur. The major mechanism of the resistant, the colicin resistant, including first is the intrinsic resistance, and second is the acquired resistance. For the intrinsic resistance, some bacteria can resist to the colicin by themselves, such as Proteus, Ridensea molecular, Serratia adversella, and Burkholderia. They involve in the part of lipopolysaccharide modification. And the acquired resistant mechanisms, it have many mechanisms, such as LPS modification at the lipid A, loss of LPS, release the capsule 1-2 trap or bind to the polymyxin and the efflux. We interest in the part of LPS modification at the lipid A because in 2016 have a emerging report about the MCR1 was detected in E. coli strain from the animal and human, and so the role of the MCR gene is the encode phosphoethanolamine transferase or PETN. This enzyme have the function add the positively charged phosphoethanolamine to lipid A of the LPS.
The moment, because there are some problems from the from Dr. Pichaya.
Were not so far a report for the human. And in this slide, I will show you the first identification by time and area. So, for the MCR1, we can detect in the E. coli that isolate from animal, human, and food. And lastly, is the MCR10 was reported in 2020 that isolate from Enterobacter and that isolate from the human. The epidemiological of the colicin resistance for the among the MCR. Okay.
Okay, now the screen is appear already.
Okay. Sorry, everybody hear me?
Yes, yes.
We have a problem.
Yeah, there's some problems from the screen to present.
Okay, and now it's okay, right?
Yes.
Okay. So. Okay. For the epidemiology of colicin resistance, among of MCR families, you can see MCR1 and MCR9 were mostly wide disseminated worldwide, and followed by MCR3, 5, and the 8 and 2, something like that. But however, have the only single report form MCR6 was found in the United Kingdom. And this slide, I want to show you the global distribution of plasmid-mediated MCR1 colicin-resistant strain that isolate from environment, food, animal, and human in 2015 to 2016. And you will see that the MCR1 were disseminated worldwide. That is found in North America, Europe, Asia, Africa, and South America. In Thailand, the first report, the first MCR1 harboring E. coli was isolated from stool specimen that collected in 2012 from the asymptomatic persons. And next year later, MCR1 harboring E. coli will report that they isolate from the patients. And moreover, MCR1 has been isolated from the 2 animals in Thailand. We found in fly and bird, but however, no data on the distribution and characteristic of the MCR gene derived from the livestock in Thailand. Don't have the data about this, so let's do our study. Objective in our study to determine distribution, molecular characteristic, and antimicrobial susceptibility test of E. coli harboring MCR1 to MCR9 isolated from the slaughter pig in Thailand. Okay. We collect the E. coli 779 isolates from the slaughter pig in 10 provinces in Thailand. After that, the E. coli will detect the MCR1 to MCR9 gene by multiplex PCR. And then the MCR positive isolate will perform the antimicrobial susceptibility test with the diffusion and blood microdilution. And the last part, positive MCR isolate will perform the molecular typing by using the MFST and the Kermon typing method. For the result, we show that we found the 61 isolate contain the MCR gene, and you will see we found MCR1, MCR9, 3, 6, 7, 8, and cohabiting 1 and 3, 1 and 9, and 3, 6, and 7. And the proportion of MCR1 and MCR9 were higher than the other MCR genes. After that, we test the MCR positive strain in the MIC, or minimum inhibitory concentration of the colicin. From this slide, we show you the MCR1, MCR9, and MCR cohabiting 1 and 3, 1 and 9, 1, 6, and 7 were resistant to the colicin. Of them is the 79 isolates that clearly resistant to colicin. And some point that interesting, the isolate harboring MCR1 show the more resistant to colicin than the E. coli harboring MCR9 from this result. So we think that maybe MCR1 may strong resistant to colicin than the MCR9. And after that, we performed the antimicrobial susceptibility test of the MCR harboring E. coli isolate. We performed 61 isolate, and the results show that MCR positive isolate were susceptible to the third generation cephalosporin 82 to 93%, and susceptible to the fosfomycin 95%, amikacin 98%, and maybe it's good news now. They're still susceptible to carbapenem 100%. And for the molecular typing for the MCR harboring E. coli, in this part, the common typing show the phylogroup A predominance, as was the predominant about the 27 isolate, and follow phylogroup B1, group D, and phylogroup B2. And moreover, after we performed the MLST, we found the most frequently of the MLST ST101 and ST8900 in 9%, more than another the ST. And we found all of the 25 ST, and we found 16 isolate as the represent as the new ST. So and nowadays in Thailand, in the another study, they found MCR 1, MCR 3, MCR 5 in the animal, maybe and in the animal, I think in pig or in pork, something like that. For the conclusion, from our study, we found the distribution of MCR gene in E. coli isolate from slaughter pig 7.9%, and we found MCR1 was the most predominant, follow MCR 9, 3, 6, 7, and 8. Moreover, the occurrence of multiple MCR gene has been found in product. MCR1 with MCR3, MCR1 with MCR9, MCR3 with MCR6, and MCR7. So from our study, we suggest that the presence and dissemination of MCR gene in the food supply chain, livestock, and environmental should be investigated. And strengthening the official surveillance system should be controlled, prevent, and minimize the dissemination of MCR gene from animal to human should be priority in the future. Okay. So thank you everybody for your attention. Okay.
Hello. Thank you, Dr. Pishanakan. Right now, I will take a question
Sorry,
from the floor. Is there any question from the floor, please?
No.
Okay, I have questions.
Thank you very much.
Thank you for your nice presentations about antibiotic resistant genes in pigs in Thailand. So because nowadays there are evidence, as you mentioned, that there are evidence reporting about antibiotics resistant genes in pig in Thailand.
Yes.
Because of using antibiotics in industry and also agriculture.
Yeah.
In case of controlling microorganisms, could you please suggest us the way or strategies to control bacterial infections in animal farm without using antibiotics? Do you have any suggestions or recommendations?
Oh. In my opinion, we should use the how to say, it's difficult to tell about the antibiotic use, right?
Mm-hmm.
Maybe some farms still use.
Mm-hmm.
Because maybe I think now the government not allowed to use this antibiotic.
Mm-hmm.
I'm not sure, but maybe. But
okay.
Farmer and worker, that's still use. So it's hard to suggest in this point.
Okay. Thank you, Dr. Pishaya.
Okay, thank you.
Okay, if there is no question from the floor, right now, as we reach the end of our program, may I invite Assistant Professor Dr. Vutinan Raksachit, Deputy Dean for Planning and Research, to deliver the closing for this meeting.
Thank you, Associate Professor Dr. Chainalong. On behalf of the committee organizations, Faculty of Veterinary Technology, Kasetsart University, Faculty of Veterinary Medicine, Rachamankala University of Technologies, Tawan Ok, Bang Phra Campus, Thailand. I would like to extend my deepest gratitude to honorable speaker, distinguished guests, and all participants for attending this seminar, both on-site and online meeting during 2 days. And also thank you, the organizing committee that contribute to high value and success of this seminar. So I hope that this seminar will serve as a window for research connection and collaborations in the future about animal health and environment. Now, I have the pleasure to officially close the seminar. Thank you.
Please, thank you very much for your speech for closing. And right now, I think this is a good time to take a picture together. Please, everyone, open or turn on your web camera to take a picture for closing the meeting, please.
เดี๋ยวขอเปิดกับตัวเองครับ
Just a few moments, please, to wait for everybody to turn on their camera. Thank you so much. We are done for today. Thank you so much, and thank you everyone for being here today. And see you next year for the first of the conference. And have a good day. Bye bye.